Dr. Kapil, I spoke to just now Mehta Saab. He is joining in after 2 minutes. He's in office.
Okay, sir. Okay, sir. Okay, sir. Thank you.
I appreciate that we could get a very big person and all.
Yes, sir. Yes, sir. Thank you. Thank you.
It is a 335 no total.
Yes, sir. 332.
And we should take his help periodically. I will connect you to him. Okay.
Sure, sir. Sure, sir.
Welcome to today's chief guest, Professor Guwahati. Also, my mentor. Someone joined or not?
Abhijit is here, so he has joined, and yeah, Sumanji came, so he'll join soon. Abhijit is here.
Don't make any noise, sir. Once started, don't make any noise in and around.
Yes, sir. Yes, sir. Yes, sir.
No question of opening the door of the conference room. Some sound I don't accept.
Yes, sir. Yes, sir.
Old one, not this one, but it's okay now. Yeah. Okay.
Good morning, sir.
Good morning. How are you?
I am doing good, sir. How are you, sir?
Well, okay. Nice to be able to participate. So good.
Thank you very much, sir, for accepting our invitation. And we are privileged, we are honored.
Yeah, you know, your director is my very good old friend. and I cannot say no to him. But as I said, I'm only formally attending it. I'm not giving any lecture or talking something any substantial. Just to reaffirm my support for all of you.
Thank you, sir.
Sir, if you permit, we can start, sir.
Yeah.
Okay. Kapil, shall we start?
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
Good morning to all of you. Welcome to the one-day virtual conference on emerging trends in medicinal chemistry, which is as a part of Azadi ka Amrit Mahotsav that is being celebrated in Guwahati for the past few months. Welcome to today's chief guest, Professor Govardhan Mehtaji, University Distinguished Professor and Kalam Anjiridi Chair at University of Hyderabad, who is personally who is my mentor and well-wisher of NIPERS in general, well-wisher of NIPER Guwahati in particular. Welcome, sir, to this NIPER Guwahati virtually. We are looking forward to see you physically. Then only my medicinal chemistry department will have a lot of confidence that we have a wonderful support from you. And also welcome to the distinguished speakers who are excellent. Kapileshwar Seth, who is the organizer of this conference. Sir, I will give you a little background about NIPER Guwahati, which is established in 2008 and which is the first national pharma institute in Northeastern region. It was mentored till 2016 by Guwahati Medical College. Ever since I joined, I have taken over that one, and then 2016 onwards, we have shifted to the temporary campus. Now we are in the permanent campus. I'm happy to say that after NIPER Mohali, NIPER Guwahati is the second institute to have its permanent campus with the 2 lakhs square feet of the total area. And also in recent NIRF ranking 2021, NIPER Guwahati could secure 19th rank, and we have 8 departments. 8th one is inducted recently by our secretary suggestion, Dr. Waghela, because medical devices is very, very important. That's why we have recently started total 80 departments. I am also happy to share, sir, this is a unique NIPER to have 8 national centers, like GLP accredited center by biotechnology, GMP accredited center by Ministry of Commerce, and the animal breeding, first animal breeding center in Northeast regions to promote the drug development through animal research. So not only for me that animal to be produced for exclusively for NIPER Guwahati, but also to the surrounding institutes of the NIPER. Information technology and electronics technology, MIT 1.5 crores, and that is also unique center. This is the NIPER connected to the National Supercomputing Mission, and we are also closely working with the CDAC for the high performance computing, not only Param Ishan, Param Padma, and Param 10,000, sir. I think Mehta Sahab, you are well aware. When I was in IACT, we have conducted joint program with the CDAC and IACT for 12 years advanced course in the bioinformatics. With that experience, I have reconnected again my NIPER to the CDAC to get the maximum benefits for the drug design through in silico research. And our main strength of this institute is we are sitting on the gold mine, as I discussed earlier over phone to you, where we need your guidance and support and mentorship. We have almost 125 compounds with us who are given by the 75 traditional healers. Some of them are literally proved for the various diseases, but there is no preclinical data in entire Northeast. That is why not only for traditional healers products, even for the government institutes like Government Ayurvedic College and all, they have got the proven, especially government institutes, they have the proven clinical data, but they cannot go to the market or they cannot see the light of the day without preclinical data, where NIPER Guwahati is supporting them in all angles. Now, we are not only doing education and research, but also societal research, especially in this direction of the drug development, especially by maintaining the quality control and quality assurance. We have very strong department of the quality control, sir, that is now headed by Dr. Radhakrishna, who has a breadth of experience in more than 20 quantifiers of the industry like United States Pharmacopoeia, Indian Pharmacopoeia, Reddy Labs, and so on. They are taking care of it. This is a nutshell, very brief about our institute, sir. And we need your support and mentorship. Once again, I welcome the chief guest as well as the welcome to the wonderful speakers of today. I congratulate Dr. Kapil Seth, who joined recently after completion of the 2 postdocs in IIT Bombay and 2 places renowned institutes in USA. Now he is the medicinal chemistry assistant professor. One more person, Vaibhav, is going to join through exclusively the computational chemistry. And this is our little bit background about my institute. And with this introduction, once again, welcome all of you. And over to Dr. Kapil Seth and invite our chief guest.
Thank you very much, sir, for your nice introduction about NIPER. So the conference has been organized to enrich medicinal chemist toolbox by thorough interaction with the well-recognized scientists and scholars in India and abroad. We are sure that the intellectual merits of their lecture will definitely guide us to move forward in a right direction, and we can sketch our roads to deliver in a better way. So now we'll move to our next session. It is my pleasure to introduce the renowned scientist, Professor Govardhan Mehta, as the chief guest.
Kapil Seth, and we will just give the mic to Professor Mehta ji. Okay?
Okay, sure, sir.
Over to Mahotsav, because I know his style and all, and he don't want to waste his time. No need of introduction for him also. Over to Mahotsav.
Okay.
Thank you.
So, good morning, namaskar to everybody. Professor USN Murthy, Dr. Seth, distinguished speakers, and of course, distinguished participants in this virtual meeting. I'm delighted to be with you this morning, although very briefly. I'm not going to talk about either my research or the great scientific strides that have been made in medicinal chemistry and likely to add further in weeks to come. I am basically here to express my solidarity with what you are doing and also express my support for your present as well as future activities. Let me first compliment NIPER Guwahati and your leader, Dr. Murthy, for excellent progress that NIPER Guwahati has made in last few years. Really very, very commendable performance. And I must also say that I have been very familiar with the idea of NIPERs, which in my view was a great conceptualization about 40 years ago. I was privy to some of, being an old person, I was privy to some of the discussions that took place when NIPER Mohali was established. And as you know that it actually was a very successful enterprise at its time. I'm happy that later on the NIPER emerged as a very well-known brand and many new NIPERs have been established. And I'm particularly happy that NIPER Guwahati has really turned out to be a flag bearer of new NIPERs in our country. So my very best wishes, Professor Murthy, and appreciation for the excellent work that you are doing. I would like to now say a few things about medicinal chemistry, because the theme of this virtual seminar is emerging trends in medicinal chemistry. I don't need to emphasize the importance of medicinal chemistry, particularly the events of last 2 years in the post-pandemic world. Contributions of medicinal chemistry has become both vital and critical. In addition to the challenges of the pandemic, as you know that there are huge challenges associated with MDR and AMR. And of course, people say that there are millions of zoonoses, the viruses, which are lurking around. And you never know when the transmission of these viruses from other species, particularly from animal species to human beings, will take place. And therefore, medicinal chemistry will have a central role in shaping the future of drug discovery and for providing succor to humankind. I just want to make 3 points about how we should be looking at medicinal chemistry and how it should be advancing in future. I will not go into the topical outlines or what are the frontier areas or what is the research area which is more promising than the other. What I intend to do is to convey to you what kind of approaches we should take to promote medicinal chemistry. And of course, if you have creative people, you show them the direction, they will deliver and find their own path. So what I'm going to share with you is 2 or 3 facilitating steps that we need to take to advance medicinal chemistry further. The first and foremost is that we should recognize that medchem is no longer a subsidiary area. You know, traditionally, for many, many decades, medicinal chemistry has been considered as an add-on to either mainstream pharmacy education or mainstream chemistry education and so on. I think we should give it up and it should brought into mainstream. I understand from the little folder flyer that you sent to me that you have a department of medicinal chemistry, but there are not very many places in India where medicinal chemistry is a separate department. In fact, at the most, people specialize in medicinal chemistry. There is a paper or a course in medicinal chemistry, but not a full-fledged department and course on medicinal chemistry, degree course in medicinal chemistry. So I think one of the steps that many of our institutions, not everybody should do that, but many institutions who have some strength and want to build up the arena of medicinal chemistry further should seriously think of mainstreaming medicinal chemistry, because we know that medicinal chemistry is a truly interdisciplinary area, and therefore it is good that it is highlighted in the activities of the institution and duly supported. I am not going into the details how we do it. It is left to the leadership of every institution. How do you go about doing this? Secondly, many of you must be familiar with this concept of One Health. You know, these days everybody talks about One Health. One Health is the concept in the well-being and holistic healthcare of all humanity in every country. So One Health is a WHO sponsored and widely accepted concept. Now, you know, and just to briefly, I'm not giving a lecture on One Health, although I'm very familiar with the area. One Health is both an interdisciplinary concept, but more important is it's a concept which is adaptable to individuals, to communities, to countries, to the whole world. And it's a very holistic idea. If I recall correctly, I think in one of the recent addresses, even our Prime Minister also spoke about the possibility of One Health. So we have to understand what One Health means, and it must percolate down. So the idea of One Health is something that we should adopt. And in an institution like pharmacy, where you have many components, medicinal chemistry is one component of One Health, but you have many other components. From your director just now mentioned about the enormous wealth of natural resources, particularly plant-based resources that you have in the Northeast region. This is very ideally suited for addressing some of the issues of One Health. Not every issue, some. Everybody cannot do everything. But you must understand and realize and recognize the idea of One Health. Unfortunately, it has not permeated in our system yet, but it should, because it's a global idea and many countries have adopted. So that's the second point I want to make. The third is we should recognize the relationship and how to make medchem or reposition medchem in the age of sustainability. I can only say that about a year or two back, there was a symposium in IIT Chennai on medicinal chemistry, and I gave a lecture there. And I will only give you the title of my lecture. They wanted me to inaugurate and give a lecture. I gave. The title of my lecture was Medicinal Chemistry in the Age of Sustainability. You know, we are living in times of sustainability. much beyond green chemistry and so on and so forth. So what are the components of medicinal chemistry which are going to be vital in this age of sustainability? I don't want to repeat the lecture that I gave, but it is important that MedChem, with regard to both discovery research, diagnostic research, and more importantly, process research, must adopt the founding basic principles of sustainability. We can no longer have, because any advance in MedChem will require manufacture even at the discovery stage, then of course more manufacturing at the clinical stage, and eventually if approvals are there, then in manufacturing. So the principles of sustainability should be guiding. I'll give you a very simple example. There was a time when people were boasting of libraries of compounds that they make. When combinatorial chemistry was at its peak and was fashion of the day, although when you look back, you know that it was not a very wise way of looking at chemistry, but chemistry also has its own fashion. When combinatorial chemistry was at its peak, there were write-up that you can make 1 million compounds in a flask using combinatorial techniques. Today, we should ask, is this the way to do MedChem? And we should also ask, is it a sustainable way of doing MedChem? So today, MedChem must therefore undergo major changes. Every compound that may have medicinal property may have to be ultimately be made, but not in very early phase it has to be made, because today we have empowerment through digital tools, through AI, ML, and many other things. And we should use all these tools before we embark on making compounds. As a synthetic chemist, I realize the importance of making compounds, but making compounds can only be a starting point if there is a huge creative element. But otherwise, you must use other techniques available to you before you embark on a medicinal chemistry related project. So this is the major three things. Remember my urging to you, restore the primacy of medicinal chemistry and center stage it, and particularly in an institution like NIPER, recognize the concept of One Health and recognize the idea that MedChem should fit into the ideas of sustainability, which are going to drive every human activity. You know, next week we have this COP26 meeting in Glasgow, and India is fully committed to the concept of COP26, and our Prime Minister is actually physically going to make a commitment to this meeting. And therefore, it is important that anything that we do must be compatible with sustainability, because the COP is about climate change, and sustainability cannot be attained without addressing the issues of climate change. So I'm sorry I have digressed a little bit away from your main thing, emerging trends, but I do believe that at a conceptual level, what I have told you are going to be not only emerging, but the trends in medical research, not at molecular level. At molecular level, many of our participants, you all are masters in manipulating molecules. I am presenting to you the conceptual framework along which MedChem should advance. So once again, all the best. My appreciation, Professor Murthy and Dr. Seth, for organizing this meeting to sensitize our colleagues and give them an opportunity to share their work and ideas with you. And all the best, Professor Murthy, to NIPER in its advance. It's already achieved very visible and commendable progress, but excellence is always a journey. So be on en route to that. Okay. Thank you very much. Thank you. Thank you.
Thank you. Thank you very much for your lucrative, informative, and thought-provoking address, sir. It will be taken care by NIPER Guwahati. And I'm very happy to share that more than 335 participants from India and across the world, they are participating now in this conference. They are all immensely benefited by your address. Thanks so once again for being with us. Namaskar.
Thank you very much. Thank you. I will log off. Kindly permit me.
Please, sir. Please, sir. Thank you. Uh, Welcome, Dr. Vishal. and
So, yeah, so let me introduce Dr. Vishal. So, it is my pleasure to introduce our next speaker, Dr. Vishal Rai. Dr. Vishal Rai obtained his PhD in chemistry from IIT Bombay with Professor INN number 3. He subsequently held a postdoctoral position and MITACS Elevate Fellow at the University of Toronto, Canada, with Professor Andre Udin. His contribution to peptide macrocycles created the platform for in-cycle therapeutics. Later, he joined the Department of Chemistry, ISAR Bhopal in 2011. So his research group is leading the development of chemical technologies for the precision engineering of proteins. He's the founder and director of Plaveltech Private Limited, the state-of-the-art protein and antibody engineering technologies such as LDM, glide tag, and mass spectra empower the company. Also, he is the recipient of very prestigious Swarna Jayanti Fellowship, Ramanujan Fellowship, CRSI Bronze Medal, CDRI Award for Excellence in Drug Research, and DAE Young Scientist Award, to name a few. Recently, he joined the ACS Chemical Biology team as an early career board member. Also, he is an invited fellow of the Royal Society of Chemistry, UK. So, with this brief introduction, I would like to invite Dr. Rai to deliver the lecture.
Thank you, Dr. Seth. Very good morning, Dr. Murthy and Dr. Seth. Am I clear? Am I audible?
You are audible.
Okay.
Yes, Dr. Rai.
So before I share these slides, in case if there is any technical glitch, I will keep this on side. Just drop me a message, and I'll just make sure that I'm back. So let me just share.
Kapil, you have, and you can share in case if you have any problem. Okay.
Sure, sir. Sure.
Okay, wait a minute. So, Dr. Seth, is it visible?
Yeah, it is visible. Please go ahead.
Okay.
Thanks.
Thanks a lot. So at the onset, let me thank Dr. Seth and Naipur Guwahati for giving me this amazing opportunity, and also to allow me to talk about the science we are doing at ISA Bhopal. Our team at ISA Bhopal is developing chemical technologies for precision engineering of proteins. And besides that, we are looking into how some of these methods can meet the technological demands in the field of biology and medicine. So what I'll do in today's talk is that I'll take some of these methods in the backdrop and will tell you a story revolving around the life of New. Now, today's lead character, New, is very much like any one of us with a balanced and predictable personality till it's alone. But things do get far more interesting when New is part of a larger social gathering. So let's assume for a moment that this lady in the pink top is New for today. Now, what you would notice is that she's very likely to coordinate her dance moves with the people who are in close proximity to her. And let's just call that as the first circle of the social network. Now, similarly, the second circle is going to affect the first one, and this is going to continue over the next circles. So whether New realizes it or not, the second and subsequent circles are also going to have a direct or indirect impact on her actions. Now, these modes of communication in the social network, they draw great similarity with what happens in the cellular network. In fact, they also are connected because the social network would have implications on the emotions, which in turn is going to regulate the generation of variety of molecules, for example, neurotransmitters that are going to then regulate the function of both mind and body. Now, what you would notice is that this is a closed loop, and the dimensions of these loops can vary, but one thing will remain constant, that the actions of nu are going to be a lot impacted by the social network in which it's present. Now, if we have a little closer look at what happens at the cellular network situation, again, this becomes reinforced by the observation of how feedback loops operate in the cellular network. You can also think about the enzymes that are fairly promiscuous in nature. So depending on which social ecosystem they belong to, their functions would be different. Now, with this little background of how a nu can be operating, let's just reimagine nu to be a small functional group in a small molecule, a collection of few atoms with the capability to donate a pair of electrons. Now, by that definition, we can also call nu as a nucleophile. So if a nucleophile is positioned in a small molecule, then its behavior, its electron donating capabilities, its selectivity profiles are going to be fairly well manageable and understandable. However, as soon as you take this nu into a relatively more complex social ecosystem, there will be a much more range of variation in the parameters. And that is something that becomes really interesting. And now you can imagine that this number of social networks will only keep on increasing as we move forward towards live cells or tissues or animals or even human beings. So in the nutshell, what we are saying is that we very well understand how a nucleophile can donate a pair of electrons to electrophile and lead to the bond formation, or how it can take back its pair of electrons and break the relationship with electrophile and lead to bond dissociation. But this is what also leads us to the most important question for today's talk, that can we understand the spectrum of nucleophiles' behavior in the complex social environment that is presented by biomolecules and the cellular system? And if we can understand it, can we take it forward to design a chemical toolbox for the precision engineering of these biomolecules and then translate them further? So if you look a little closely, it is challenging landscape of reactivity and selectivity, but since it is fairly unexplored, it also provides an excellent opportunity to create new dimensions in the field of not just chemical biology, but also therapeutics. So with this little background, if we look into the molecules of protein as organic compounds that are full of nucleophilic residues, then if we have to do a covalent modification, typically we will be adding an electrophile. So a nucleophile is going to be reacting with that electrophile, but then it will also have all sort of competitors that are going to be present in the solution. So let's just assume for a moment that this nucleophile is a lysine or a primary amine in general. So the electrophile, if it can react with a primary amine without touching any other functional groups that are nucleophilic in nature, that is what we define as chemoselectivity. And once we have such an electrophile, it would realize that, okay, there are multiple copies of primary amines that are going to be present in the protein. So for precise single site modification, it will have to figure out how to differentiate one primary amine from all its other cousins that are going to be present in the protein. And that is what I'm going to term as site selectivity. Now, if you look into these primary amines, out of the whatever number is present, one of them will typically offer a little high reactivity in combination with good solvent accessibility. Now, we call such reactive centers as reactivity hotspots. So labeling a reactivity hotspot is one aspect, but the next question is that can we label this primary amine in this case or this one? So can we go beyond the reactivity order defined by the protein, and can we have such technological platforms? And that is where I'm going to bring in the term of modularity. So these are few jargons that I'm going to use in the upcoming slides, and that's why I thought that I'll give you a little backdrop on this. Now, at this point, it is also a good question to ask that, fine, this is challenging and all, but then why do we even need such methods? So the understanding of reactivity and diverse landscape of selectivity would allow the capability for developing tools for both biophysical chemistry and biochemistry. And the fact that the non-selective tools themselves have a huge margin am I connected?
Yeah, you are connected. Dr. Rai, you are connected.
Okay, because it just gave me a message that there was some disconnection. Okay.
Oh, now it is good. Please go ahead.
Thanks. So, so while we are looking into how we can address some of these demands, the main focus of our research group is in a different problem that is related to address the technological demands in the field of directed cancer chemotherapeutics. And we want to do that by synthesizing homogeneous antibody drug conjugates. Now, as the name suggests, this state-of-the-art platform involves a monoclonal antibody that is linked to a drug or a toxin molecule covalently with the help of a linker. And now once these ADCs are given to the patient, the job of the antibody is to go forward and target the antigens that are overexpressed in the tumor cells, but not in the normal cells. And that's the basis of differentiating the tumor cells with the normal cells, and that's where directed component comes into the play. So based on this platform, the 14th drug got approved earlier this month, and they are all available for the treatment. But there's one thing that is very common to most of the drugs in this segment, that they are sold as heterogeneous mixture. And by that, I mean there will be 0 to n number of toxins attached per molecule of the monoclonal antibody. Okay, now this heterogeneity, it's fairly well established that it is detrimental to both pharmacokinetics and batch-to-batch consistency. But not much can be done in this regard simply due to lack of the technologies that can address the question. Now, if you move forward, the exact same problem is also bogging down the fields of conjugate vaccine, where instead of an antibody, you have a carrier protein, and instead of a toxin, you have antigen attached to it. And this percolates further in the field of imaging-guided oncologic surgery or photodynamic therapy that is done with the help of the antibodies. Now, our team has been working in collaboration with a few other teams across the globe to see if we can bring in protein-based therapeutic solutions and surgical solutions. But while we are working on this segment, our long-term dream is to see whether we can precisely target a single site of a single protein inside a human body. But the point is that if we can do it, then we are looking at a disruptive platform for precision therapeutics, because then small molecules can probably deliver the specificity that has so far been possible only with biologics, and that would be amazing. But before we can learn how to do this with complex systems such as humans or tissues or even cells, we would first need the knowledge and methods to regulate the reactivity and selectivity with isolated proteins. And once we have that, we can translate it back to the more complex systems in attempt to get to the technological solutions for humans. Now, at this point, it's a logical question to ask that do we not already have drugs that are highly specific in nature? So let me give you one example to put things in perspective. Kinases are very well investigated because they are involved in several critical signaling pathways in humans. So it's not really a surprise that a lot of pharma companies have been focusing on developing kinase inhibitors. In fact, there are around 38 approved drugs in just last 1 decade. But if you look into the best protein kinase inhibitors that are out there, they go for at least 30 to 100 known targets within the proteome, and then there are multiple unknown ones that are not identified. Now, that's not surprising because that also explains why there are side effects in the patients who administer these drugs. And if we are successful in our endeavor, we would be able to address some of these technological demands. So with this question in mind, we started with very first question in this direction, that let's see first that if we can develop methods that can do precise single site modification of native proteins. But before we can address this big question, we bottle down to a relatively smaller question, which really involved to see whether if we can have chemical methods that are capable of identifying the reactivity hotspots and label them with very high efficiency without compromising the precision. Now, we had to actually tackle a few other questions before we can even reach to this answer. Specifically, the question of concentration disparity. So most of the organic chemistry reactions are developed in the millimolar concentration range, which is great for small molecules, but if you have to work with proteins, you just can't operate beyond micromolar concentration. So if you bring down the concentration of the reaction, it will definitely take care of the protein aggregation problems, but then it also compromises the kinetics. So the bioconjugate chemists tackle this problem by taking large excess of electrophile, and that does take care of the kinetics component, but then it also comes at the cost of selectivity. So to address this question, we came up with a very simple solution that if we can design a molecule that has 2 electrophilic centers such that the nucleophile from protein could attack on the first one, generate a latent nucleophile which can then react intramolecularly with another electrophilic center to give the final product. So if we can shift the rate determining step from an intermolecular process to an intramolecular process, then the overall method would be independent of the rate of the concentration of the substrates in the medium. And that is what we exactly realized with the help of N-hydroxythalamide, which allowed us to label the reactivity hotspot, which was at the N-terminus alpha amine of the protein. And not only it works in the case of RNase A, it also applies fairly well to many other structurally diverse set of proteins. So with this in hand, we moved on to the next question, where we wanted to see if we can differentiate the reactivity of a nucleophile from its multiple copies. And for multiple reasons, we wanted to do it with the lysine and the primary amine that is there in the side chain of the lysine. But just now we also discussed that N-terminus alpha amine offers higher reactivity than the lysine-based epsilon amines. So we had to figure out how to block it, even if transiently, to even try to address this question. And after a set of screening, we realized that redefining the reactivity landscape on the surface of protein actually helps a lot in this perspective. So if we treat a protein with an aldehyde, we can convert this nucleophilic surface into electrophilic surface. The only difference is that here the imine at the N-terminus can react with the penultimate amide bond intramolecularly to give imidazolidinone. Now, this is a highly reversible reaction, but it is good enough to block the reactivity of N-terminus imine for the external nucleophiles that would be added in the second step. And that creates the opportunity to see if imine formed by one lysine can be differentiated from the others. And after a lot of screening, we figured out that copper slide does a great job in single site modification of lysine in proteins. But if we want a metal-free version, phosphomanic reaction does a great job where triethyl phosphite is added as a nucleophile. Now, the targeting of reactivity hotspot is not limited only to lysines. It can go forward and we can target even histidine residues with this structurally minimalistic electrophile, which is cyclohexanone. So simple reaction between imidazole of histidine in a Michael addition fashion gives this adduct, and the balance between reactivity and compromise of selectivity is such in this case that it allows us to do single site modification with pretty high efficiency in a structurally diverse set of proteins. And once the job of the Michael acceptor is done, it renders this carbonyl group, which can then be utilized for installation of diverse probes. Besides, you can also think of utilizing the pKa value of this alpha proton and think of inducing a retro Michael addition to release or dissociate this carbon-nitrogen bond in a chemically orthogonal fashion. So you can take it forward for doing metal-free protein purification. So let's say if you have a recombinant protein, in this particular example, UBC9, that has his tag at one of the terminus, then you can treat it with simply cyclohexanone. The reaction is done, and once it is over, you can simply pour it to the hydroxylamine functionalized resins, capture the labeled recombinant protein with his tag, remove everything else, and simply induce the retro-Michael addition with the help of imidazole buffer in a chemically orthogonal shape to get metal-free purified proteins. Okay, so this is one interesting method for specifically purifying the proteins that have to be used for the therapeutic purposes. Now, whatever examples I discussed till this slide, they all have been targeting reactivity hotspots. And it was time for us to see if we can go beyond that, if we can go beyond the protein-defined reactivity order and target unique residues in the protein and hopefully in a protein that is residing in a cell system. Now, we also were aware that to address this demand, we will have to simultaneously take care of multiple challenges. So we came up with an idea that we like to call as disintegrate theory in our lab, where the point is that we can use multi-step chemical processes to deconvolute the challenges of reactivity and selectivity, and hopefully that can provide us a better regulatory tool to define what we want from the chemistry and not the transformations that are actually defined only by the protein and limit us in terms of what we can do in engineering of those proteins. So let me give you one simple example which would elaborate what I'm talking in this slide. So the first example that we discussed with thalidomidation or N-terminus alpha-amine modification, there when we add an electrophile to the protein, that single step has to take care of both chemoselectivity and site selectivity. Now, in the second case, when we were talking about the modification of lysine, we added an aldehyde that basically delivered a chemoselective transformation to generate imines. And now the second step, which is giving the irreversible modification, has to worry only about site selectivity. So chemoselectivity and site selectivity are deconvoluted in two different steps, and that makes it far more easier for us to regulate the precision. Depending on what goes wrong, you know which step you can tune. Okay, now once we did that, we realized that we can take such intermediates, and if somehow we can generate another reactive intermediate in a reversible pathway, it will just open up completely new dimensions of selectivity. So let me show you one successful example that emerged from this theory. So if we take glycine, one of the simplest amino acids you can think of, treat it with this aldehyde with a proximal hydrogen bond acceptor, this very special aldehyde leads to the formation of amino alcohol through this carbon-carbon bond formation. Now, let's see how it works. This amine and aldehyde would react first to generate an imine. Now, the proximal hydrogen bond acceptor would stabilize and facilitate the generation of this enol type of intermediate, which in turn attacks on another molecule of aldehyde to give amino alcohol, and subsequent hydrolysis would just render this final product, which is the amino alcohol. Now, what is even more exciting is this fact that if R1 is anything else but hydrogen, the steric constraints would not allow this activator to come in the close proximity, which means this nucleophilic intermediate is not going to be generated in such cases. And what it translates to is that the glycine is going to render very high efficiency of this amino alcohol formation, and every other natural amino acid which is substituted is not going to give any irreversible product. So we took this N-glyce specific technology and demonstrated that we can modify a diverse set of proteins with N-terminus glycine. In fact, the fact that you can express recombinant proteins with N-terminus glycine in a pretty straightforward fashion, it just opens up a lot of scope for translation of this method for biochemical engineering of proteins. And once we were able to demonstrate this, it was time to move forward and redesign the reagent a little bit. So we made this symmetrical GlyTag reagent with both the ends doing similar job. And in this example, if we take insulin, which has two N-terminus, two chains, one of them is N-terminus glycine, and that is what reacts with this GlyTag reagent, leaving one more aldehyde at our disposal with some unreacted insulin. So if we take this whole mixture, pour it over acyl hydrazide functionalized resin, everything that has this aldehyde would get captured, and you have single site immobilized resin, which can be very well stored in the freezer for a very long time. And we also recover the unreacted insulin and use it in the next cycles. Now, as and when required, we can take this immobilized insulin out and just do a transoximization reaction with the probe that is functionalized with hydroxylamine, and that reaction would deliver analytically pure single site tagged proteins. Now, this state-of-the-art method simply delivers the bioconjugates of 100% purity level. So if we take one of these conjugates, for example, this fluorophore tagged insulin, and we test it in a particular pathway, then we also find that the binding of such modified insulin to the insulin receptor or its downstream activity in terms of phosphorylation of one of the downstream protein, they typically remain unaffected. So with this, we were all set to see how we can translate this method to a more complex ecosystem of proteins. So we took a mixture of 7 proteins in which one of the protein had N-terminus glycine, in this case it's insulin, and we demonstrated that the method allows us to label just single site of a single protein without touching anything else in this reaction. So this was quite encouraging for us to take it forward and see how it is going to perform if we are working with a mixture of few thousand proteins that cell lysate has to offer. So we took a cell lysate which had overexpressed SUMO1 with N-terminus glycine. Oh. Connected?
Yeah, Dr. Rai, it is connected. Absolutely fine. Please go ahead.
Oh, okay. I'm not sure why it is giving these messages again and again.
Oh, I think Professor Mankar has joined. So, ma'am, please. Yeah, ma'am, please switch off your video. So
I will. I just wanted to make sure that I'm able to see and hear everything. That's why. Hi, Vishal.
Hi.
Hi, good morning, ma'am. So yeah, you are. We can see you, and you are audible, ma'am.
Hey, Vishal, can you just turn off yours once? I will also see if mine is getting shared or not getting shared. Or you have already.
So I'm in the middle of talk. Can I finish it?
Okay. Sorry then. I'm sorry.
Okay. So now, while we were trying to translate this method to mixture protein, the next step was to see whether we can translate it all the way to the cell lysate. And in this case, we took SUMO1 that was overexpressed with the N-terminus glycine. And it was really pleasing to see that not only we can target this specifically, it's just the N-terminus glycine that is modified. And then we can take forward all the translational steps of installation of a probe, purification, and everything to make it really a strong platform for delivering precise single-site modification of proteins in a cell lysate and tag it with whatever probe of interest we want to. Now, for a lot of downstream applications, it was important for us to see if we can have a chemically orthogonal platform for dissociation of this carbon-carbon bond. Now, let me just show you in a different figure. So what I'm saying is that the question was whether we can simply dissociate this carbon-carbon bond, and that will give us the native protein back. But then it turns out that this bond is pretty stable, and it was not really possible for us to dissociate in presence of variety of additives, including bases. So finally, when we were able to figure the polarization of this electron density in the carbon-carbon bond, that gave us the solution to this problem. For example, if we take something like PLP or pyridoxal 5' phosphate, this aldehyde allows us to generate the imine, and the extended polarization of electron density that is enabled by this additional heteroaromatic ring ensures that the electron density at this carbon-carbon bond is sufficiently polarized so we can dissociate it under physiological conditions, very mild conditions, to release the GlyTag reagent, and upon happening that means that basically allows us to get the intermediate, which upon subsequent hydrolysis would release the native protein, which in this case is again insulin. So with both GlyTag technology and retro-GlyTag technology in hand, we took the combination and we moved forward to see if we can address the technological demands of various prospects. But let me just share one example in this perspective to you. We prepared a GlyTag resin, and this is something that is now being used by several members in the community of biological sciences, where they can simply take a recombinant protein with N-terminus glycine in a cell lysate, pour it over the GlyTag resin, and that simply allows the capture of the protein of interest. This is something that can be stored for a very long time, keeps even unstable proteins stable for a considerably long period of time. And as and when required, you simply have to treat it with a PLP-based buffer, and that releases the pure protein for the next set of application. So with this, let me move on to the final question that I want to discuss today. And that, as I promised, was related to development of modular technology to precisely label the single site in a native protein. Now, here we wanted to go beyond the reactivity order defined by the protein. And we knew that it is not going to happen till we keep targeting single sites in the protein. So we had to engage the social ecosystem of the protein for delivering this job. And let me show you how we did that. So in this example, I have highlighted lysine in blue color and histidine in red color. So the idea was that if we can target the pair of residue, then we will be able to utilize the social ecosystem of a particular residue, and that will be something that can create the difference and the final judgment in terms of what gets modified. So the way we did it is by designing a reagent that had two functional groups, FK and FH, and here FK was supposed to give me a very rapid reaction with lysine that is reversible on demand, and it has to be chemoselective for lysine. Whereas FH was supposed to give a relatively slow reaction that is irreversible and chemoselective for histidine. So if I can make such a reagent and treat the protein with it, the FK is going to go and rapidly react with all the lysines that are accessible, and then I can regulate the concentration of FH near a particular histidine depending on how I design my spacers. Now, there's one more important point to see here. The desired selective pathway goes through an intramolecular route, whereas any undesired non-selective reaction has to go through intermolecular route. And that's where we have advantage in terms of kinetics by more than 1,000 times. So we took this forward and demonstrated with myoglobin that we can label histidine 36 or histidine 81 sitting in completely different domain simply by positioning FK and FH in three-dimensional spec differently with respect to each other. And this method does not work only with myoglobin. We have translated it to a diverse set of proteins and for modification of different residues within each of those proteins. So once the job of the linchpin is done in these methods, it is free for further manipulations. So you can utilize this aldehyde that is left over here for installation of variety of probes of interest, or even better, you can utilize it for capturing on the resin, purify and recover the unreacted protein. And then again, as we discussed in the previous case, it is accessible for us to give us analytically pure single site tagged proteins. Okay, so at this point, with LDM platform that was modular, established and working well in our hands, it was time to see if we can make homogeneous antibody drug conjugates for the translational purposes. Now, before I get into those details, let me just give you a quick recap of the challenges that are associated in this particular segment. Okay, so as we discussed in the beginning, the antibody drug conjugates are comprised of three key components. One of them is going to be the toxin, which in this example is monomethyl auristatin E. The second component is this antibody. And then finally, the third component would be the linker that is going to connect the toxin and the antibody together. Now, if you notice carefully, this is where the first question would come, that how do we regulate the homogeneity of this bioconjugation? And once that is figured out and the ADC is injected into the bloodstream, the next challenge would come in terms of bond strength that is connecting the toxin to the antibody. Because if these toxins are released before reaching the target, then you can expect that they are going to show off-target activities and they are going to inhibit the normal cells along with the tumor cells. And that is something which has to be regulated by the chemistry that we would be using in the process and the regulation of the bond strength. So if these two parameters are also taken care of, eventually the ADCs are going to reach the tumor cells where the antibody has to bind to the antigen. And as you would notice, the conjugation site becomes so critical because it should not affect the binding process of the ADC with the antigen. And if it is properly done, then the endosome is going to be formed with the complete internalization of this whole complex, where eventually it will take it to the lysosome, where the lysosomal degradation over here would release the toxins, which then can lead to cell death and apoptosis. Okay, now this process constructs the first and one of the pathways of how ADCs lead to the killing of the tumor cells. Apart from that, there are two more pathways that are associated in this whole process. So the ADC binding to the antigen also ensures that the antigen-mediated signaling pathway is inhibited. Apart from that, it also takes care of extracellular domain shedding process, and it inhibits that in the complex situation. Besides, one of the very important segment is to see that how the FC region of the antibody should also be left unperturbed, because this is going to engage the immune system by taking the NK cells cooperating to it and leading to the antibody-dependent cellular cytotoxicity. So this gives you a clear indication of why it becomes so essential to regulate the precision at different levels. And once you start looking into the various modes that can be responsible for the resistance in these cases, it becomes even more evident that why it is so important to come up with a very comprehensive technological platform for construction of ADCs that not only regulates only homogeneity and drug to antibody ratio, but also allows you to control the chemistry, the residue of conjugation, the bond strength, the site of conjugation, and the linkers. And that's exactly what we are trying to deliver with the LDM platform. So let me take one example from the first case where we selected trastuzumab as the monoclonal antibody and emtansine as the toxin. And the reason why we picked this up was deliberate. We wanted to do a head-to-head comparison with Kadcyla from Roche. So what you will notice is that the LDM-derived ADC gives a great efficiency in inhibiting the antigen-positive cells. And not only it outcompetes Kadcyla, but more importantly, it just shows no negative impact on the antigen negative cells. And that's the specificity that it is offering in comparison to antigen positive tumor cells versus antigen negative normal cells. Now, in this particular case, the conjugation site is histidine, but the LDM platform does offer us great versatility. So we can take it forward and utilize it for engineering lysines precisely. And then let me show you one example from the library of ADCs that we made using this platform, where we install the toxins at lysine number 169 and lysine number 395. And this ADC again does a great job in inhibiting the antigen positive cells, outcompeting Kadcyla, and more importantly, just not showing any negative impact on the antigen negative cells. So I hope this convinces you that these platforms, the LDM platform in particular, provides one of the best opportunities to create library of antibody conjugates and meet the technological demands in the field of directed cancer chemotherapeutics and guided surgeries. But moving forward to one more concept that I mentioned in the beginning, if you just look at the LDM platform, how it brings in the specificity, if there are around 100 competing sites, the linchpin allows you to bring it down to around 10, and the design of the spacer ensures that you can modify a single site in the protein. Now, this is fine with the isolated protein, but if you go towards the cell lysate or cells, you would realize that now in a eukaryotic cell, you have around 20,000 proteins that are going to be present. So your competition went up by almost 20,000 times. Now, how do we design linchpin at this stage so that the number of competitors come down to at least a level where spacers should still be capable of bringing in single site modification? And that is one thing that our group has been looking into for the past few years. And I will not be sharing this part of the story in today's talk, but definitely probably at the next occasion. So I hope I was able to convince you that our discoveries in the past 10 years, which allows us to control the reactivity, chemoselectivity, site selectivity, modularity of different types, and protein specificity, it does provide a very comprehensive toolbox for precision engineering of native proteins, assists us in coming forward with protein-based therapeutic solutions, and hopefully will allow us to address precision therapeutic solutions in the future. Besides protein specificity, we have also been able to demonstrate how we can go forward with organelle specificity or cell and tissue specificity. But these are the stories that I would definitely be glad to share at another occasion. Besides these things, our group has also been trying to take the knowledge that we are gaining from in understanding the reactivity landscape of protein to see if we can design chemistry with small molecules to create a new domain altogether. Along with that, our couple of group members in our lab have been constantly trying to solve the problems related to one of the biggest bottleneck, which is analysis, unambiguous analysis of the exact site of conjugation, the degree of conjugation, and they have been able to do some wonderful job in this direction, which is also helping our ADC candidates that we are trying to take forward towards the preclinicals. So with that, it is time to thank all my group members who have done the actual job in the lab. We are really thankful to all our collaborators who have been constantly supporting us in various dimensions of the science related to what I discussed today. And we are really thankful to all the funding agencies for believing in our research program and generously supporting us over the years. So thank you all for your kind patience. I'll be glad to take your questions.
Thank you very much, Dr. Rai, for your insightful talk. Now the session is open for question and answer. So participant, please feel free to ask questions. So dear participant, please feel free to ask any questions if you have any. So, Dr. Rai, I am having one question. Can you go to that slide where there is kind of your imine type of concept? I forgot the slide number. Sorry.
It's fine. I'll remember. Yes. Wait. It is. This is the one.
Yeah. This is the one. So here you are making that amine nucleophile. Amine is a nucleophile, so you are converting this amine to an imine species, right? And yeah. So now that imine, you have explained that imine has now become electrophile, right?
Right.
Okay. So yeah. So can you please explain this slide a bit more? So I couldn't follow you in that time.
So what happens is that if let's say if you're trying to target a protein at a single site, right?
Yeah.
So the general reactivity landscape is nucleophilic, means you only have nucleophilic residues on the protein. So if you will be adding an electrophile, it will be target. So it already has a reactivity order. Whether you're choosing a soft electrophile or relatively hard electrophile, you can have chemoselectivity for, let's say, cysteine or for primary amine. But if you're going for something like primary amine is a high frequency residue. What that means is that for most of the proteins, the number of primary amines would be very high. Also, it is charged in the physiological condition, which means the number of solvent accessible primary amines would also be very high. So in a way, the number of competitors are very high. So now the idea was that, okay, protein as a nucleophilic surface has one reactivity order. But if we add a reversible reaction to it, such that we generate a different reactive intermediate, so in this particular case, it is electrophilic, then the reactivity order might not remain same. That was the idea that we were trying to explore. And this came as a bonus that the N-terminus imine was something that got protected, and we were able to characterize this in some control experiments. And then we realized that, okay, this is what allows us to block the N-terminus, and then so this is not reactive anymore. So one of the imines from a lysine would then react.
Right.
Now, again, what I did not mention is the number of cases that were explored that did not give selectivity. So a lot of cases, what happens is that the reactivity of the nucleophile is too high. If it is not on the subtle side, then it is going to, by the time it gives 5%, 10% conversion, the second site would also start getting it will start participating in the reaction. So single site modification can be achieved till, let's say, 5 or 6% conversion, but beyond that, you will just have second site also getting modified. So these are two selected methods where you, for example, in this case, you can go up to 99% conversion of single site modification before a second lysine based imine will start participating.
Okay, so that in that case, the imine, I think you made the imine in situ in the reaction medium. Am I
Right.
correct? Yeah, because imine is kind of unstable. So I worked on this kind of chemistry as a transient directing group and tried to functionalize kind of CH activation with the metal. So we tried to isolate the imine component, but it didn't work. I mean, imine was unstable to isolate. Yeah. So,
Yes.
but
So it means obviously it means they are transient, and especially when you're working in water, you can see that even the reversibility factor is going to push it more towards the starting materials. And from if you worked on CH activation, then you know this directing group concept, the proximity effect concept.
Yes.
That you utilize in CH activation or functionalization. It's not very different from, at least philosophically, it's not very different from what we do in case of proteins.
Right.
Especially in the linchpin directed modification, you see, it was like a proximity control where there was a lysine group directing the modification of histidine.
Yeah. So it's pretty much interesting. So I'll discuss with you at some point later. Yeah. So thank you very much. Dear participant, do you have any question? Please feel free to ask.
Or if they want to ask later, they can very drop an email to me.
So if you do not have any question, please join me to thank Dr. Vishal for his insightful talk. A big applause to Dr. Vishal Rai, and thank you for accepting our invitation. Thank you once again.
Thank you very much.
So now we will move to our next session. So our next speaker is Dr. Prathama Menkar. So it is my pleasure to introduce Prathama is Menkar. So she is currently working as senior principal scientist at CSIR Indian Institute of Chemical Technology. She is chair Department of Organic Synthesis and Process Chemistry. She has experience in various stages of drug discovery program due to her association with the industry and academia. Dr. Prathama's research activities encompass an excellent blend of medicinal chemistry plus process chemistry using modern organic synthetic tools. Her contribution in developing HDAC inhibitors, peptide mimetics, carbohydrates, green synthesis of drug-like molecules, and drugs are highly commendable. The contributions of Dr. Prathama resulted in discovering of molecule which completed to phase 2A clinical trial. She is a team member of the total synthesis of the largest man-made drug, eribulin. She has approximately 80 publications and 3 patents against her name. She has received OPPI Woman Scientist Award in 2016 and Best Woman Senior Scientist Award, Genesin Educational Impression Award in 2021. She is a fellow of Telangana Academy of Sciences. With this brief introduction, I would like to invite Dr. Prathama to deliver the lecture. Ma'am, please.
Thank you, Kapileshwar. I'll try sharing my slides. I'm sorry, Vishal, I didn't realize it was in the middle. I thought mine was at 11:20. So you are also trying is the feeling I got.
So it was actually, we are running a little bit, early. So it's no issue, ma'am. Please share your slide.
Are they taking time? I don't know what went wrong.
Okay. So, ma'am.
Yeah.
So, ma'am, click on that up arrow that is share the content.
It is going in.
and select entire screen.
I will, but it is still going in round and round.
Okay, ma'am. So, yeah.
Do you see anything?
Ma'am, we can't see your slide.
I know, it's just stopping at a certain stage. I don't know.
Sharing full screen didn't work that well for me, so I then chose the other option where I was able to share only PowerPoint window.
Yeah, but that also is not happening, unfortunately. It's just going. Sudarshan Chakram is turning. This online platform, it's not happening, Kapileshwar. It's stopping. It's not allowing me to share. I'll stop my video and see if that helps. Kapil, I think what I'll do is I'll log off and log in. Maybe that will help. Okay?
Okay, ma'am. So, WhatsApp নেই, এমনি কল করো না। এমনি ফোন দেবে না তো? বাইরে গিয়ে এয়ারটেল নম্বর থেকে এটা।
Okay, let me try again. This is not cooperating. Okay, Kapil, quickly what I'll do is I'll send my slides to you. Okay?
Yeah, ma'am. That is fine. That is fine.
I'll just send my slides to you. Because otherwise it will never happen. Okay, I just sent my slides to you, Kapil.
Ma'am, just now you sent, so in my Naipar Guwahati email.
Whichever mail you have been sending me.
Okay, right. So
I'm sorry.
it's no issue, ma'am. Yeah, ma'am.
So if you can run, maybe that will help me because it's not happening.
Yeah, sure. Yeah, ma'am, we got your slide. So
Yeah. Thank you so much. I don't know what the mess is, but anyway.
yeah, ma'am. Is it now visible?
Yes. Thank you so much, Kapileshwar.
Thank you. Ma'am.
Thank to all, and I sincerely thank Naipar Guwahati team, including Dr. U.S.N. Murthy, for giving me this opportunity to share some work that we do in the area of drug discovery. So the basic question is, I'll tell you when to change the slides, okay? So yeah, go to the next slide, that's fine. So the basic question that in today's pandemic we have been thinking, and before these 2 years, we were like sure that we have conquered the world and we are able to manage the diseases. And then suddenly the pandemic struck us, and then the question of why drug discovery has become a irrelevant question, because now we really know that we have to do drug discovery. So I'm starting with this, that why do we have to do drug discovery? So there is always unmet medical need where there are new infections that are being detected now, and SARS-CoV-2 has taken us for really a big spin. whatever we knew so far, at least we had to unlearn it and learn new things. Then Ebola is another example where we have not been able to find a cure. Then there are other molecules which have been approved as drugs, but we have found them to be having low efficacy, and they are especially in the cases of CNS disorders where dementia and other diseases, the drugs have to be given in larger quantities because their efficacies are low. Cancer is another area where we have many drugs approved, but they don't have the kind of efficacy that we would desire to have. They are working, but not to the extent it's desirable. Then many of these drugs are known to have unwanted side effects, especially in the case of antidepressants, antipsychotics, and antibiotics is antibiotics or anti-infectious area is one area where we have seen a lot of side effects and resistance developing because of the side effects. So that's antibiotics, antivirals, antimalarials. These are the areas where not many new drugs come in, and whatever come, there is a quick appearance of resistant varieties, and then the work has to start once again. In addition, there is a cost of medicine which needs us to look for new molecules or new drugs. Monoclonal antibodies, just like Vishal had given a talk on how to modify them, but they come into the category of slightly higher cost medicines which are not affordable to most of the people. Then there are some medicines which have to be taken for a lifelong time of a person, and they contribute to the expenditure of the individual as well as the country, and these come into the category of depressions or metabolic syndrome, especially diabetes or hypertension, where people have to take the medicines for their life, and the individual or the country which bears the medical cost, because in many of the Western countries the medicines are sponsored by the country. There is a lot of pressure on the medical system that the cost have to be more affordable. In addition to that, as I am a chemist, I would rather like to look at the sustainability of the processes that are developed for the molecules, and that's also an area where we need to do drug discovery. So we need to have better scaffolds, because all these points that I noted earlier, efficacy or side effects or resistance appearing, and in addition to that, we have to develop drugs which will have as low dosage as is possible to have the effectiveness as we desire it to be. And sustainability of the raw materials is something that is a high concern right now, because we are depleting many of our resources so fast that we may not be able to get a lot of compounds later as we progress further, and we will be leaving the planet for the next generation without any access to those chemicals which we have been using extensively. Please, next slide, please. So after finding out why drug discovery, I would slightly do a history work on how old is drug discovery. So as long as, I think, from the time humans came on this earth, they started looking for finding a cure of any of those things that affected them. There were initially plants and minerals that were available that were used as drugs, and then Indian medicinal system is something that elaborately noted down the effect of many plants and minerals. Egyptian also is a very well studied medical system. Chinese and Greek were also there, and then so all the civilizations basically had their own systems of finding a cure for the diseases that were prevalent at that time. And also, as the times changed, they also started merging with the later advances in medicinal chemistry. Next slide, please. So what are the sources of drugs? As I mentioned, they can be plants. There are many cases of animals giving us clues about finding a new molecule which will work, and snake venom is one of those areas where the cure for, the maintenance of blood pressure could be found out because of snake venom, because in Americas there was a snake bite would result in dilation of the blood vessels, and people were able to find out that this is because the snake poison contains a compound which dilates the blood vessels, and so that's how the beta blockers and management of BP was taken up extensively. Minerals have been used in Greek medicines. Minerals have been used extensively. Even mercury has been used in Chinese medicine. And marine flora and fauna have been a rich source of new scaffolds for us to find out new molecules for various applications. They are not limited to one or two applications, because many antivirals, anti-cancers, or even now for SARS-CoV also, maybe there'll be something that will come out of marine flora or fauna. And synthetic chemistry has been one of the major contributors to the drug discovery and sources of drugs. Next slide, please. So this is the process through which drug discovery goes, and most of you would know this, but this is just a pictorial explanation of. So how does it start? It starts with identification of a target, then validation of it, then identifying a molecule which will work the best on that identified target, then identifying and optimizing the hit to the lead. Then we do the preclinical studies, then go to the clinical studies, then the drug authorities approve the molecule, and the molecule comes into circulation as a drug. Next slide, please. So this is a typical funnel that we always see, 10,000 molecules giving rise to, if we are lucky, one molecule which goes into the market as a drug molecule. And the process is complex, complicated. It has all the chances of the drug or molecule not going to become a drug. And then how do we overcome is the exciting journey of drug discovery. Next slide, please. Drug discovery needs all the disciplines that we can think of to come together to make a molecule into a drug. So we have chemistry, we have biology, we have computer sciences. So everybody has to contribute to make sure that a molecule from showing some activity to becoming a drug and being available to the population for finding a cure. So this slide basically tells that all of us are equally important for finding a drug molecule. Next slide, please. So what I'm going to cover in my talk today is the ways of finding a molecule which has the potential to become a drug. So there are basically 6 approaches that I'm going to cover here. There are many more, but I'm going to cover 6 approaches here. So natural product based approach is one. Computer aided approach for finding a molecule is another one. Target based is the one that I had described earlier, that you find a target and then find a molecule which will fit for the target. Epigenetics is something that is emerging as an important area of drug discovery. And SARS-CoV-2 has taught us that repurposing is an area we should not neglect, because it had gone on a back burner for some time now, but now it has again got prominence, because whatever molecules we got today for treatment or management of COVID-19 are repurposed molecules. We don't have a new molecule nor drug for treatment of COVID-19. And AI-driven applications are also there for finding new drug molecules. Next slide, please. So David Newman has been writing articles on how much of natural products contribute to the drug discovery and finally get approval from the drug authorities. And this is a pie chart which shows that roughly around 50% of the molecules have either natural product as a scaffold, or they are natural product derivatives, or they have a pharmacophore which is a natural product. There are synthetic molecules which is maybe 30%, and remaining are all biologics and other molecules. Botanical drugs are also making their presence felt, and this is a fairly large collection, because it covers 1981 to 2019. So this has been the trend that natural products have given a large chunk of scaffolds for finding cure for many of the diseases. Next slide, please. So these are some of the molecules which I just wanted to show, because being a chemist, this is what excites us more than those pie charts and everything. So salicylic acid is one of the smallest molecules which we can think of, and which has been there in use for now around 150 to 200 years. Penicillin is always a hero molecule, because we all wish to have a molecule like penicillin, which will change the life of others as well as change our life. Then azithromycin is there. Paclitaxel changed the treatment of cancer. Cyclosporine for when the organs are given, when there is a when the organs are replaced and the body tries to reject them, and cyclosporin is used to suppress the rejection. Rapamycin has been a molecule which is again it is turning out to be an Amrit kind of molecule because it has applications in many diseases, and more is being found out about the applications of rapamycin. Next slide, please. Eribulin is something that I just wanted to show because this is an exciting molecule. If you look at the parent molecule, helicondrin, which I'm showing here, it is such a big molecule that synthesis of this molecule was a nightmare for any chemist. I mean, it's a challenge, but it is also a nightmare because you have so many centers, you have so many rings, and they have to be in a particular way to be having the activity. So ISAI has done a great job of finding out which part of the molecule is giving to the activity. And from helicondrin, next slide please, we get the molecule eribulin. So this has become a very small version of helicondrin which has the best activity, and this molecule has been shown to have very good anti-cancer activity, especially in cases where people have been given the first line of treatment and they have developed resistance for it. So, and in metastatic breast cancer cases. So eribulin has been a molecule of choice, and further applications of this molecule are being explored. Next slide, please. So I'll go to our experience in finding out hits and some leads in these areas. So first is natural product based molecules. Next slide, please. So this is a very small molecule that we started working on. It's called sulforaphane. It is found in cabbages and all the crucifery family members like broccoli, Brussels, and cabbages. This molecule has a very unique isothiocyanate group, but if you look at it, there is not much of the scope for doing SAR on this molecule because it is such a small molecule, and the activity is because of the isocyanate group. Next slide, please. So what we have done is we have taken one part of the molecule at a time and tried to modify it and see what was the activity of it. So since it's a aliphatic chain, then we synthesized compound 1. We then, because we wanted to make it more stable, we used an aromatic ring. Then we wanted to see if the SO bond, SO part of the molecule is making any difference to the activity. Compound 3 was synthesized. So this is a list of compounds that we synthesized to see what was it that was giving activity to the molecule. Next slide, please. So isothiocyanates we found out are important, and they are involved in the cell cycle arrest. They have anti-cancer activity, antimicrobial and antifungal potential. And also they are being explored for treatment of autism disorder and rheumatoid arthritis. So this functional group was something that interested us a lot. Next slide, please. So based on the molecules that we synthesized, they were screened for their anti-HIV activity. And one of the molecules, HIV 381, OCL 381, showed very good activity, and it was almost on par with lamivudine. Lamivudine had 97% inhibition, and our molecule showed 94% inhibition. But the activity was 3 times more compared to the parent molecule sulforaphane, and it is inhibiting viral replication in low as well as high viral load cases. So it is not that it is only when the load of virus is very high that it will work. It will work only also when the viral load is low. So this has given us a hope that we can work and explore this further for other viruses as well. Next slide, please. And this is the SMAR1 is the protein that is being explored, and that is the one that is found to bind to our molecule, and it has shown a very good activity. It is a immunomodulator protein, and it can have different applications in any other infections as well, in addition to being any antiviral molecule. Next slide, please. So that was the case study of a very small natural product that we developed into a molecule which showed anti-HIV activity. This is a larger molecule which is a terpenoid, and glycerinetic acid is found in most of these Southeastern Asian countries and in our country as well. And it is a abundantly available natural product, and it is used as a taste enhancer, especially when bitter drugs are given. Then this is given as a flavoring agent so that the bitter taste of the drugs is masked. And it is known to work in peptic ulcer and it has expectorant properties. Next slide, please. So what we tried to do was, this is something that has come up very well because Kobayashi has developed a very mild reaction condition to insert benzene into active moieties. So what we have done is we have taken glycerinetic acid. Can you go to next slide, please? And then by activating it as a beta ketoester, we try to add benzene onto it. And with the use of the solvent selectivity, we were able to get either a C-arylation product or a ring expansion product. And both these sets of compounds were screened for their anti-cancer activity. Next slide, please. So this mechanism is for chemists to understand that in cases where we can either the 4-membered ring opens up and then we get the ring expansion, or in cases where it gets protonated, the C-arylation happens and the ring expansion is not taking place. Next slide, please. So 2 molecules that we had synthesized, one which is C-arylated molecule and the other one in which we had done the late stage functionalization of the methyl to make it into a sulfonamide. Both these compounds showed very good activity in breast cancer cell lines. And it was much better than the parent molecule as well, because glycerinetic acid worked at 80 micromolar and our molecules worked at 5.6 and 6.9 micromolar. Next slide, please. The additional advantage of these molecules is that they were not allowing the cancer cells to undergo metastasis, and that's a very big observation that we have, and we were very excited to have it, because the main problem with cancer cells is if you try to treat them at a place where they originate, and when they migrate to the other organ, they become resistant to the drugs and persons cannot find cure for that. So our molecules at low and high concentrations are able to minimize the migration of the cells and are not allowing metastasis to happen. So the orange in the first, on the left hand side, the orange one is the one where you can see these are the images of the mice, and the orange area shows the metastasis, and then as our compound is given, we don't see any metastasis. And in the right hand side figure, you can see kidneys have high concentration. That is because the kidneys are eliminating the compound collected, and the metastatic cancer cells also are eliminated through kidneys. So when we take an image, we can see that the kidneys are showing concentration of the cells, but other parts of the body are not affected. Next slide, please. We also found out that this molecule works in the mitochondrial level, and that is another added advantage for us. And this is something that is coming up very fast. If you treat mitochondria, then maybe we can find a better cure for cancer treatment. So our molecules are working very well on mitochondria. Next slide, please. So when we looked at the metastasis, and then we realized that our molecule at high concentration, the concentration of metastatized cells has decreased a lot, except in kidneys, as I said, because that's where the cells are eliminated from the body. So we are very excited about this result, and we plan to take this forward. And we have found a use for a natural product which is abundantly available. By doing a slight late stage functionalization, we are able to get a molecule which has the potential to become an anti-cancer drug. And because it comes from a natural product, the chances of it having toxicity are reduced a lot. Next slide, please. So I go to the next one. It's computer-aided drug design. Next slide, please. So this is an area which has, in the past 15-20 years, has taken a lot of importance. It depends on the availability of 3-dimensional structures of the target proteins, and it explores the binding site cavities. Then it helps in effective and fast identification of lead molecules, and also helps in the optimization which is taken up, which is happening now, because computer-aided optimization of lead molecules is being done on a large scale. Now many companies are doing this to make sure that the time is saved for finding out a lead molecule with the desirable properties. So this uses structure-based virtual screening also, and molecular docking and molecular dynamics has been a mainstay of this kind of drug discovery. There are many molecules which have come up through SBDD, and amprenavir is one of those molecules. Next slide, please. What we have done is, before the SARS-CoV-2 hit us, we were trying to explore different molecules for finding out a cure for chikungunya as well as dengue, because these are the two areas where not many drugs are, basically there are no drugs available, and this affects our country a lot, and many people suffer because of both chikungunya and dengue. So chikungunya is because both of them create a lot of problems for persons who are affected by it, and then deaths are there, as well as people are incapacitated for a longer time, and that puts a burden on the families where these infections occur. Next slide, please. So what we have done is we have a collaboration with National Institute of Virology, and they have used the chikungunya mature envelope complex and found out we have a library of molecules in National World Bank. So we had shared the structures with them, and based on the structures, they found that some flavonoid compounds are showing very good binding with chikungunya mature envelope complex. And this actually interested us because flavonoids have not been explored extensively because they have a lot of liabilities, and they are Michael acceptors, and they have not been explored. But we found that taking flavonoids for especially chikungunya and dengue viruses is showing very good activities. Next slide, please. So Garcinia mangostana is a plant which grows extensively in the eastern and western coast of our country, and the fruits are available seasonally. And these are the different flavonoids that are present in them: alpha, beta mangostines, alpha, beta, gamma mangostin, gartenin, and garcinon. And when the binding studies were done, we found that alpha mangostin has a very good binding potential, and it has shown very good scores as well. Next slide, please. So based on the binding scores, of mangostin was done for chikungunya infection. This was done in NIV. ISCT does not have the facility, so I just don't want to say that it was done at ISCT. And then it was found that the infection, there was a reduction in the virus, and then 100% reduction of the virus at 8 micromolar concentration. And because it's a natural product, we have not done any modification on it, and the natural product is being consumed regularly. It is part of the food in the western part of the country. So it does not have any toxicity issues with it, and it can be used at higher concentrations as well. Next slide, please. Chikungunya as well as dengue both were screened for this molecule, and it was found that the chikungunya before treatment and after treatment and co-treatment, in all the cases, alpha mangostin was able to control the viral load at 8 micromolar, even as a prophylactic. Next slide, please. Same studies were done with dengue virus, and here also we got 100% reduction of the virus titer. Next slide, please. And you can see that mangostin reacts with dengue protein as well, and it suppresses viral replication in different stages of the viral replication. Next slide, please. The third area I'm going to cover is target-based. Next slide, please. So this is the work that we have done for a kinase inhibitor, and there are around 538 protein kinases encoded in human genome, and for those kinases, 38 inhibitors have been approved so far. So imatinib, elotuzumab, and there are a lot of NIBs that have been approved by drug authorities in various parts of the world. These small molecule tyrosine kinase inhibitors have been approved for treatment of non-small cell lung cancer as well. Especially that is one cancer which has not got a proper cure so far. Afatinib has been approved in 2013. There are many antibodies also which serve as kinase inhibitors, and natural products like resveratrol and quercetin are also known to work as kinase inhibitors. Next slide, please. So this is the work which we had done with a German collaborator, and the target was tyrosine kinase inhibition. After doing molecular modeling studies, we found that ureas are working as better target molecules, and it was affecting the fibroblast growth factor receptor FGFR1 and working on the small cell lung cancer. The molecule that I have shown over here, BGJ398, it has worked on patients who had a stage 4 squamous cell lung cancer as well. We synthesized a lot of analogs around this, but found that the parent compound was much better than the analogs that we synthesized. Next slide, please. So squamous cell lung cancer is it is insensitive to chemotherapy because by the time the drugs pass all the barriers and reach the lungs, their efficacy is gone. Mostly surgical resection is the one that is done, and it has a strong history of correlation with tobacco. So smokers have a larger chance of getting lung cancer. Next slide, please. So this is typically how we have done the synthesis, and we have this has been published in Cancer Discovery. Next slide, please. So the molecules were screened against first xenograft models, especially for looking for FGFR1, and then it was observed that the tumor size decreases on the application of the molecule 15 mg per kg at the concentration of 15 mg per kg. Next slide, please. And this is the this is in case of a patient. This was given to humans as well, and as you can see that the tumor size decreased after taking the molecule BGJ398. So it was given to a patient and it worked in a patient as well. Next slide, please. So I'll come to the next one, which is epigenetics. Next slide, please. So epigenetics is something that has come up of late in drug discovery, and I mean there were molecules which were known to work on different enzymes and proteins, but the idea of epigenetics being a concern or a major area of work has become of late in the parlance of drug discovery. In epigenetics, the change is in the phenotype without any change in the genotype. So the cells change physically, but there is not much of a change in the genome. There are three types of families. One is a writer enzyme, which is second one is a reader enzyme, and the third is an eraser enzyme. These are the three major enzymes being studied in epigenetics, and these introduce, recognize, or remove DNA or histone modification. So HDAC is one of the major areas in which this epigenetics has been explored. But the problem in epigenetics that is being faced today is that it needs to have a isoform specific inhibitors, because most of the molecules that have been approved so far have a pan enzyme isoform inhibition, and that creates a lot of side effects, and that also increases the dosage of the molecule. So what we have been working on is to find a molecule which will be HDAC isoform specific inhibitor. Next slide, please. So our work started way back where we had synthesized molecules which are azaflavones, and we found that these molecules, with the use of organocatalysis, which got a Nobel Prize this year, was able to give us selectivity in the compounds that we've synthesized. Next slide, please. And there is another set of compounds that we synthesized were triazoles. These we had synthesized using a combinatorial way, and then synthesized a big library of triazoles also. Next slide, please. So these molecules we had screened using Drosophila as a model, in which MIR14, which corresponds to the human MIR4644, was introduced into the Drosophila, and then it was observed that compound was able to decrease the concentration of the HDAC so that the breast cancer cells could be controlled. This was done in Drosophila. Next slide, please. So instead of stopping at that stage, we took the molecule further, and then we started checking its activity through molecular modeling and then in vitro studies. And we found that these molecules have shown very good activity. We have synthesized a library of around 150 compounds, and then there are some structural modifications which I cannot share here, but one of the molecules has shown very good activity, which we are taking forward. It's called ISCT 108. We have done in vitro studies also on that, and very soon we are going to file the IND. The advantage of this molecule is that when we were studying its activity, we realized that it is working on the idiopathic pulmonary fibrosis, a disease for which there is no clear reason for getting the disease, as well as there is no proper cure. But our molecule is working at very low concentrations on this molecule. And after the SARS-CoV-2 pandemic started, there were cases of people having pulmonary fibrosis. So we are screening our compound against the cell lines where post-COVID PF is observed, pulmonary fibrosis is observed. And once we get those results, we'll be able to file for IND. Next slide, please. So these are the data that we have generated for the molecule, and we have done against the most of the HDAC isoforms to find out whether our compound is selective or not selective. And we found that our compound is selective against HDAC 8. Next slide, please. So this is where we are right now. We have done animal studies. We have to do some large animal studies and then go for clinical trials. Next slide, please. So I come to the last part of my presentation, which is on the repurposing of drugs. Next slide, please. So how does repurposing happen is by identifying new therapeutic uses for old, existing or available drugs. And it is an effective strategy because most of the data for the drug has been generated, and taking the molecule forward saves a lot of time as well as money. Next slide, please. So this is how the molecules repurposing happens, that a molecule is known to work on a target, but a new target can be identified for the molecule, or in some cases the target is known, can be explored for another disease. Though it's known for one disease, it can be explored for another disease. So this is the two approaches for drug discovery. Next slide, please. So metformin is a classical example that has been used for treatment of, it's a maintenance disease for type 2 diabetes, but it is also used for polycystic ovary syndrome. And now it has been found out that people who take metformin could increase their lifespan can be increased. So it's kind of turning out to be elixir of life. Next slide. And in case of Ebola virus, concoction of HIV drugs which are approved for HIV treatment were found to be effective against Ebola. So azacitidine, amiodarone and tenofovir combination was found to be effective against Ebola. So this is another good example of molecules which can be repurposed. Next slide, please. When the SARS-CoV-2 started, then the problem that was faced by all of us was there were no drugs available. And so a lot of molecules were explored, and in this case we found out favipiravir to be effective. Favipiravir is an RNA-dependent RNA polymerase inhibitor, and it was meant to be an anti-influenza treatment drug, but now we found that it is working. It was repurposed for treatment of SARS-CoV-2. So ISCT worked on this molecule in a record time of 23 days. We developed the technology for this, and this was transferred to Cipla. So when the first wave hit, we could help industry to take up manufacturing of this molecule. Here we have not done any drug discovery. Drug discovery was done by others. I'm just telling what we have done. Next slide, please. So this is this is what we have done, because at that time there were no raw materials available, and there was a paucity of human resources, chemicals, solvents, everything. So whatever was available in the local industry, we used it to make the process for favipiravir. Next slide, please. In addition, we have developed a process for remdesivir, which is reducing the number of steps as well as using the chemicals that are available indigenously. This is again a classical case of repurposing of a drug. Next slide, please. These are the key intermediates, and we have developed processes for all of them, and also shared the information with industries so that they can take it up. Next slide, please. So I come to the last one, and that is AI driven. Next slide, please. So artificial intelligence has been helping and has been explored both for finding out better drugs, and how does AI help? It helps in drug design. It helps in finding biospecific or multi-target molecules. That's another area which is coming up, because when in epigenetics, when we are talking of one enzyme, if the drug can work on two different enzymes, then that is also a desirable property, because then the disease can be controlled by targeting two different enzymes. AI is also helping in chemical synthesis. AI help has been taken for drug repurposing, but now drug screening and preclinical trial and clinical trials are also being driven by AI. So these are the points that AI has been used extensively, because it's bringing down the cost. It is also bringing down the time that is required for finding a new drug molecule. Next slide, please. These are other advanced techniques in drug discovery, which I did not cover, but they are being used extensively. One is QSR. Then organs on a chip is something that has been used now. Instead of sacrificing animals and then finding whether the molecule is working well or not, organs on a chip are being developed in the laboratories, and the molecules are being found to if they are working on the molecules. Then with the 3D printing, human organoids are also being able to print and use them in the lab to find out whether the molecule is reaching the organs, and then whether it is working on the disease that has affected the organ. Then genomics and proteomics has been used, but it is being used more and more now. Then virtual trials is something that clinical trials are going towards, where many of the properties can be studied by virtual trials rather than doing physical clinical trials. Repurposing of existing drugs. SARS-CoV-2 has given us a huge chance to explore the drugs which we had put on a cold storage, because they may have the potential to treat future diseases. And gene editing technologies have also been considered now as a treatment for many of the genetically inherited diseases. Next slide, please. So with that, I thank CSIR for giving me the opportunity to do research in an area where I like to work. And this is our campus, one of the greenest campuses and a very beautiful campus. Though we are a chemical lab, our campus is very, very beautiful. So all of you are invited to come to ISCT and visit us. And I'll take up any questions, Kapileshwar.
Thank you, Dr. Pradhama, for your very much insight.
Is it over? Did I overshoot? Was the time okay?
No, it's fine. It's fine, ma'am. It's fine. So we are running a little bit early, so it's absolutely fine. So thank you very much, ma'am, for your insightful talk. And as a synthetic organic chemist, I understand the difficulty to synthesize that huge kind of molecule in kind of total synthetic pathway, in particular when there are several chiral centers. So I really appreciate your work on that area. Thank you very much. And now the session is open for question answer section. So dear participants, if you have any question, please feel free to ask.
Yeah, Vishal.
So if there are no other questions, then I can go ahead with one question that I had.
Ask me easy questions, okay? I'm not a teacher.
So my question again, I was really fascinated to see all these dimensions that you have been covering is really exciting. And I also keep thinking about how, the synthetic chemistry, the synthetic biology, and so at the DNA level, RNA level, and then coming to proteins level. So we have been mostly targeting proteins for all these years. And the gene editing and the RNA-based therapeutics with reason coming up, and then the synthetic biology further coming up, where we are using viruses as delivery agents or bacteria as delivery agents or for curing certain purposes.
Yes.
Now, and then chemoproteomics coming up to tell us that all these years we have been looking into libraries and exploring them, and yet the best leads that we have, they are mostly non-specific.
That's true.
And it's not very surprising, because if you're looking at, let's say, 5 or 6 or 7 non-covalent interactions driving a binding, its a specificity in the biological milieu is never going to be in single digit, right? So,
exactly.
But at the same time, when people who are working from the biologics perspective, which includes me as well, then they criticize, the fact that, okay, here is the non-specificity. Probably they miss one point, and that's where I want your opinion. That the fact it doesn't have to be 100% specific for one target, especially when you're going with non-covalent interaction. So how much of non-specificity is okay in general, where the body will just take care of it and flush it out after a while?
This is specific, I guess. We will not be able to give a number per se, but that is something that has been explored a lot now, because especially in epigenetic studies, they're trying to make the molecules which are not specific for one isoform, because that is creating toxicity.
So even protein tyrosine kinase inhibitors I was looking at, and the chemoproteomics results that are either coming from this big Max Planck team in Germany, or the other one that is sitting in US, and especially the pharma companies, but they don't disclose most of the results in publications. So the results primarily are showing that they are fairly non-specific, and but having said that, they have been doing a decent job for all these years.
That's
right?
True.
So, which is something that tells you that, okay, body still has a recovery mechanism, and also questions that when we are going for chemoproteomics approach, where we are connecting, let's say, an electrophilic warhead to convert a non-covalent ligand into a covalent binder, then probably the structure activity relationship is not telling everything, and we are over telling how many binding sites are there.
Yes.
What
we
you
need
also
to know a lot about how these things work. We are just, I think, scratching the surface sometimes, I feel, because the more you read, the more you think that, oh, I don't know anything in this area.
Completely agree. But one of the things that I hope you would also find fascinating is the evolutionary capabilities that comes from the biological systems.
Yes.
And when I see directed evolution being incorporated with the cellular machinery using bacteria, viruses, designing their plasmids, it's like, you can create a library of millions of compounds, and then you can screen and select one of them and evolve only that, or for enzyme for only that. That's, I hope synthetic chemistry can reach there sometime. I mean, it's even close.
We are trying. That's an area which is very interesting, and we are taking it up.
So thanks a lot. But this was really fascinating talk. I'm so happy that you guys were able to contribute in these difficult times as well. So congratulations.
It was really something that it's an experience by itself. It is a different talk, but it was like every day evening we would know that the reaction is not working. So our library work would start in the evening. Morning we put up a reaction. afternoon take the NMR or mass and see that the reaction has not worked. And the industry was like literally putting guns on our head. You said you will give in 15 days.
I totally understand.
It was available. Students had left home for their home, so it was something that we look back and now we feel very proud of it. But yeah, that was something which was very difficult.
Thanks. Looking forward to seeing you sometime in person, hopefully post-COVID is almost.
Sure. Yeah. Yeah, Kapilesh.
Ma'am, we are having few questions. So one of the student has asked, how can we target 2 or more enzymes by one ligand?
Students had left their home, so it was something we look back and now we feel very proud of it. But yeah, that was something which was very difficult.
Hello, ma'am.
My voice only I can hear. Okay. See, that is something that is being done using computer and artificial intelligence, where we want to selectively target more than one enzyme.
So one of the students has
It is not done. It is not done, but
how
it is.
can we target 2 or more enzymes by one ligand?
It is possible because they are. Now we feel very proud of it. It's running my recording. It looks like. My voice only I can hear. Okay. See, that is something that is being done using computer and artificial intelligence, where we want to selectively target more than one enzyme.
One of the students
It is not done. It's not done, but
how
it is.
can we target 2 or more enzymes by one.
Okay. I think somebody was playing the recording. Am I audible now?
Now it's okay. Earlier it was playing
Okay.
the whole thing.
So the question was that how can we target more than one enzyme? It is possible because the binding sites are mostly similar, and we can do it. And that is what we are. That is the next stage in epigenetic drug discovery, where they want to target more than one enzyme. Today we don't have a molecule which does that, but that is where the research is going on.
Yeah, so, yeah, ma'am. Now am I audible?
Somebody is playing the recording again. I don't know. The binding sites are mostly similar, and we can do it. And that is what we are. That is the next stage in epigenetic drug discovery, where they want to target more than one enzyme. Today we don't have a molecule which does that, but that is where the research is going on.
So thank you, ma'am. If any participant has any question, please. Yeah, ma'am. Now am I audible?
Somebody is playing the recording again. I don't know. The binding sites are mostly similar, and we can do it. And everybody, first you mute everybody. Then whoever is talking can switch on their mic. I'll mute mine and then I'll listen. Yeah, the question, Kapileshwar.
Now am I audible?
Somebody is playing.
I think it's at the host end.
Whoever is talking can switch on their mic. I'll mute mine and then I'll listen. Kapileshwar, I think the questions are difficult to understand what the questions are. You can send them to me and I'll answer them.
So so ma'am, the question was how can we target 2 or more enzymes by one ligand? It's giving same problem. Yeah. So hello, ma'am. Is it clear now?
I think it's at the host end.
No. Whoever is talking can switch on their mic. I'll mute mine and then I'll listen. Going in the background. Kapileshwar, I think the questions are difficult to understand what the questions are. You can send them to me and I'll answer them.
So so ma'am, the question was how can we target 2? So hello, ma'am. Is it clear now?
Yeah. No. Whoever is talking can switch on their mic. I'll mute mine and then I'll listen. Showing in the background. Kapileshwar, I think the questions are difficult to understand what the questions are. You can send them to me and I'll answer them.
So ma'am, the question was how can we target?
So hello.
Yeah, Kapileshwar.
Yeah. Now. Yeah, I think we have overcome the issue.
Hello, ma'am. Is it clear?
No, same problem. Okay, ma'am. So.
Showing in the background. Kapileshwar, I think the questions are difficult to understand what the questions are. You can send them to me and I'll answer them.
So ma'am, the question was how can we target?
So, dear participants, if you have any question, please you can email Dr. Prathama for your questions. So she will be happy to answer your questions. So
I would have to answer here also, but there is some echo, so I don't know how to handle that. Now I think it's better if you can ask me the question.
Okay, ma'am. So the question was means how can we target 2 or more enzymes by one ligand?
So that is based on the artificial intelligence studies that are being done. Because so AI-based studies have helped us in finding out ligands which will work for more than one enzyme, which will help in finding using less concentration of the molecule, like quantity of the drug will be less and it will have better efficacy. We don't have a molecule per se right now, but there are molecules which are being starting clinical trials in this area. Is this audible, clear, Kapil?
Yeah, so it was a little bit audible. So
AI-based studies have helped us in finding out ligands which will work for more than one enzyme, which will help in finding using less concentration of the molecule, like quantity of the drug will be less and it will have better efficacy. We don't have a molecule per se right now, but there are molecules which are being starting clinical trials in this area.
So
is this audible, clear?
Yeah, so it was a little bit audible. So
AI-based studies have helped us in finding out ligands which will work for more than one enzyme. Okay, then anything else that?
So please join me to thank Dr. Prathama for his insightful and wonderful talk. A big applause to Dr. Prathama.
Thank you.
Thank you, ma'am. Thank you.
Okay, then anything else that?
So So now it is time so that we'll move to our next speaker. So our next speaker is Professor Indrapal Singh from IIT Madras. Sir, can you hear me?
Yes, I can hear you.
So
Are you able to hear me?
yes, sir, we can hear you, but we cannot see your video. Please switch
Yeah,
on.
I will start that.
So our next speaker is Professor. So sir, before starting, let me introduce. So it's my pleasure to introduce next speaker, Professor Indrapal Singh. So Professor Indrapal Singh obtained PhD degree in chemistry from the.
I can hear you.
So
You are able to see me on the video now?
yes, sir. We can hear you, but we cannot see your video. Please switch on.
I will start that.
Sir, let me introduce. So yeah, so Professor Indrapal Singh obtained his PhD in chemistry from the Department of Chemistry, University of Pune. Following his PhD work and a brief stint of National Chemical Laboratory, Pune, as fellow scientist, he went for his.
Are you able to see me on the video now?
Yes, sir. We can hear you, but we cannot see your video. Please switch on.
I will start that.
Sir, let me introduce. So yeah, so Professor Indrapal Singh obtained his PhD in chemistry from the Department of Chemistry, University of Pune. Following his PhD work and a brief stint of National Chemical Laboratory, Pune, as fellow scientist, he went for his.
Are you able to see me on the video now?
Yes, sir. We can hear you, but we cannot see your video. Please switch on.
I will start that.
Sir, let me introduce. So yeah. You, but we cannot see your video. Please switch on.
I will start that.
Ah, only. So, sir, we cannot see you. So you are not visible.
No, I'm switching it on now. Right.
Yeah.
I hope you are able to hear me and also see me.
Yes, sir. Yes, sir. It is absolutely fine. So let me introduce. So it's my pleasure to introduce Professor Indrapal Singh as the next speaker of today's event. So Professor Singh obtained his PhD degree in chemistry from the Department of Chemistry, University of Pune. Following his PhD work and a brief stint at National Chemical Laboratory, Pune, as fellow scientist, he went for his postdoctoral studies at University of California, Santa Cruz, USA, and where he worked in the field of radical mediated reaction. Later, he moved to the University of Constance, Germany, as an Alexander AVH fellow to carry out research work with Professor Richard R. Smith's group in the exciting and challenging field of synthetic carbohydrate chemistry. Later, he joined the Department of Chemistry, IIT Madras, as an assistant professor in 1995 and rose to the rank of full professor in 2006. He later served the department at IIT Madras as head during the period of 2015 to 2018. His research interests lie in the area of synthetic carbohydrate chemistry, synthesis of biologically and medicinally important compounds, developing enantiopure chiral building block, etc. He has published 86 publications in peer-reviewed international journals of high impact factor and filed 4 patents. So Professor Singh is the recipient of several prestigious awards and recognitions: Alexander Von AVH Fellow, JSPS Fellow. He was elected as member of National Academy of Science, Allahabad, Excellence in Carbohydrate Research Award, ACCTI, in 2 consecutive years, 2014 and 2015. So with this brief introduction, I would like to invite Professor Singh to deliver his lecture. So please, sir.
Thank you for the introduction. What's your name?
Kapil, sir.
Kapil, good. Nice to see you because I have not seen you, and therefore I asked you this question. So thank you very much for the invitation and also this opportunity to give this lecture. So I will share my I hope you all are able to see my slides.
Yes, sir, it's absolutely fine. Please go ahead.
Just 1 minute. Yeah, I'll make it full screen.
Sure, sir.
Yeah, I think you all are able to see the slide.
Yes, sir. Yes, sir.
Yes. Good. You want me to keep the video on or would you, because of the internet connectivity and all, or would you like me to close the video?
So, sir, you can close the video and you can deliver the lecture. It is fine,
That's
sir.
great, because it's only for the internet point of view I'm saying. Sometimes when
Yes,
the
sir.
video... Okay,
yes, sir,
good. Thank you. Okay, now, the title of my talk, as I said, is Natural Products and Medicinal Chemistry Inspired Synthesis in the Recent Years. Now, I request my younger audience, particularly the student community, to just keep focused on natural products, medicinal chemistry, and synthesis. These are the 3 keywords based on which they'll be able to get most of the information which I would like to pass down to them during this 45 to 50 minutes lecture. Okay, before I start the lecture, I would like to just 1 minute. Yes, this is the building from where I'm giving my lecture, just to get you all connected. And this is a beautiful campus where you can see deers moving around. And this picture I've just taken a few days back, how nice the campus looks like. This is just to give you a feel where I am. And this is a very, very beautiful campus, worthy of visit at some time. Okay, now let me go to my talk. The talk, basically, I said, says about natural products, medicinal chemistry, and synthesis. And I'll definitely illustrate to you with 3 examples what humble contributions we made with taking all these 3 keywords together. Natural product is known since, I think, ages, centuries. And you can see this just graphic picture which tells you it started with herbal preparations where quinine, colchicine, codeine, salicin, way back, see, it is 1825, just go only 200 years back. But mankind was using natural products right before Christ also. I will not go into the history, but it's very fascinating. But if you see, as the chemistry advanced, isolation of natural products became extremely important because many scientists started feeling that natural products have a concoction and it's better to identify the active principal ingredient which is responsible for the drug-like properties. And this resulted in the growth of the natural product field. But unfortunately, during last 2 decades, there was a lull. And there was a reason for it because other streams took up very faster. So there was a period where interest in natural products seem to have gone down. But the best news is that after 2015, when the scientists William Campbell and Omura, along with you were given this Nobel Prize, and this again started seeing re-emergence of natural products. So I can confidently say that now the scientific community is once again heading towards this natural products as a valuable source, not only from plants, but from other sources also for the lead molecules. Now, if some of the students are interested in just seeing this, they should go and see this very wonderful article written by David Newman and Gordon Craig. This is 2016. I'm sure after that, few more reviews have come, which are worthy of seeing how natural products are again re-emerging as a very, very valuable lead identification area. Now, just to illustrate the point that natural products are, in fact, directly there in the society, you can see the first example. This is your Taxol, which is being used in the hospitals as an anti-cancer compound. Patients go, just get the injection, intravenous injection. Again, after 1 month, another dose. And this has been extremely popular and very effective drug as for the cancer. If you go back to this old molecule, again, very interesting history how it was discovered from the soil around this old castle. Students can definitely enjoy seeing its history. Doxorubicin is again another anti-cancer drug. And again, I'll not go into the details of it because it's only to tell you that natural products have been there and they have continuously contributed to medicinal chemistry since several decades. This is another molecule. See on the left-hand side, myriocin. It's a fungal metabolite, not coming from the plant world, but from fungus world. Seemingly very simple, that inspired and led to synthesis of fingolimod, which is also called as FT5-720. And in fact, this was the drug approved for multiple sclerosis just, I think, 7-8 years back. And in fact, we have also given a very good synthetic scheme for this molecule, where we'll be able to change the polar head group at will. So there lies the advantage of good synthesis program. Now, this is another natural product from sponge. It's a nucleoside, and this led to the discovery of this cytarabine, again an anti-cancer compound. So in short, I have just given you a few examples showing that how natural products inspired the drug discovery process. Now, this is the final example. You can see the complexity of the molecule. Helicondrin B, again isolated from sponge, but it led to a very good interaction between 3 institutes, Harvard, NCI, and SI, and led to the discovery of eribulin, again a very valuable anti-cancer lead molecule. And this is the last one, epothelone. The same story goes. It was isolated as a natural product and led to the development of ixabibolone from Bristol Myers Squibb, which is currently under clinical trials. Now, the natural product keyword is over. Now you come to the second keyword, that's medicinal chemistry. Now, medicinal chemistry students will definitely realize that this is one of the fields which is highly multidisciplinary. I will not use the word interdisciplinary, but it's multidisciplinary. And you can see Alfred Berger, who wrote several volumes on medicinal chemistry, has very elegantly pointed out that it's heavily interdependent, but independent. And these words remain even true today. And it depends on several fields, which I have written here: biochemistry, molecular biology, pharmacology, analytical chemistry, computational science, and you can add even few more, which may not be there. In short, it's multidisciplinary. No one can do medicinal chemistry alone. It has to be in a team. That's the key word. Now, what happens is, what is a drug? if you ask this simple question, drug meets the target and produces the clinical effect which we want. But now where are the targets? Obviously, if you see the current time through which we are going, it's all coronavirus. So we are looking at the virus. Virus is responsible for the ailment. So we are talking about pathogens. But no, you may have the targets also in the humans. When we are not dealing with any infection, we are dealing with something else like diabetes. No, it's not because of pathogen. It's because of something metabolically something had gone wrong. So obviously you can have targets either in humans or in pathogens. Now keeping this in mind, what are the targets structurally? And I've just listed for the sake of completeness, you have enzymes as targets, membrane receptors as targets, nuclear receptors which are in cytosol or a nucleus, you have ion channels, nucleic acids which could be DNA or RNA, structural proteins, transporters. This list may not be the complete, but I think most of the targets are listed here. Now enzymes, everyone knows you have different kinds of enzymes. I'll again not go into the details of that because that's not the objective. I just want to give you the feel that there are different targets for drug discovery. Now, see this one example. I've intentionally put enzymes. Now, in this slide, you see there are two molecules of acetyl coenzyme A at the top, and you can see that your acetyl coenzyme A on the left-hand side is your enolate form, and the right-hand side is the keto form. And there's an enzyme E1. This just does a nucleophilic addition to a carbonyl group. What will it lead to? It will lead to the formation of this beta keto ester. Now, another molecule of enolate, again enzyme E2 catalyzes and adds on to the carbonyl carbon, not to the ester carbon, ester carbonyl carbon. Students will be able to appreciate that, why it happens, and leads to the formation of this molecule, which I've shown on the right-hand side. Now, please see this molecule very carefully. One side there's an ester functionality, the other side there's a the carboxy functionality. And now what is it called? It's a 1, 2, 3, 4, it's a 3-methylene between 2 carboxyl, that's glutaric acid. No wonder why it is called as, at the beta position you've got methyl hydroxy, it's hydroxy methyl glutaryl coenzyme A. I have read it intentionally completely because the compound which inhibits this enzyme, which enzyme? HMG-CoA reductase. Now, why I'm describing all this is the beginning of the scheme synthesis of cholesterol. Cholesterol is made in the body by almost 30-35 steps if we count each one of them. Every moment our body is making cholesterol somewhere. And this is the beginning of that. And you can see in the beginning stages itself, if we can inhibit this enzyme, HMG-CoA reductase, which does the job of reducing this ester functionality to this alcohol, you will be able to block the synthesis of cholesterol. And in fact, this, what I said, has been done and has led to the discovery of statins in the market, atorvastatin, which is multi-billion-dollar drug in the market. And this is nothing but enzyme inhibitor. That means your target was enzyme, and it is inhibiting and bringing about the clinical effect of lowering cholesterol production in your body. Now, another quick example of another drug target, you can see ion channels. Now, I have intentionally written only one example of lidocaine, such a simple molecule. It can be done at class 12th. It's a reaction of an aniline with some acid, beta amino substituent acid. It's an amide functionality, simple molecule. But what it does, it's a local anesthesia. Whenever you want to bring about some small surgery, it's a local anesthetic, just give this few milligrams injection. It just blocks the ion channel. Which ion channel? Sodium ion channel. And stops the nerve impulse going from the site to the brain, and the sensation is lost. So this is a very good example of the target being ion channels. It's such a simple molecule. Again, I repeat, drug meets the target and produces a clinical effect. Now, the clinical effect here was loss of sensation, and it did the job by reaching the target. Which target? The ion channels. And which target? And precisely an ion channel. It is the sodium ion channel. I can just keep going on, but that will take away lots of time from me for what I have to say about the three examples which we have taken care of. But students can note this and they can themselves take this as an exercise. The G protein coupled receptor, what is the drug in the market? Ligand-gated ion channels, what is the drug in the market? Tyrosine kinase linked. All these targets will have their own drugs. Here I have again written, if you have this transporters being inhibited, you have omeprazole, you can again see its mechanism of function. Basically, we were trying to see what are the targets for the drugs in the humans and in the pathogens. Okay, that completes the medicinal chemistry just introduction. Now, I'm coming to exactly where we focus. Now, I said enzyme is the target. Which enzyme we focused on? It's aldose reductase. Then we focus on another receptors, which are PPARs, peroxisome proliferator activated receptors, extremely valuable for diabetes-related complication. And then finally, transporters as targets. Which transporters in precise? It is sodium glucose linked transporters, SGLT. So you can just remember now the keywords are ALR, which is aldose reductase, PPARs, which are nuclear receptors, proteins, and then transporter SGLT. Now, all these three things have to come together. What are those three keywords? Natural products, medicinal chemistry, and the synthesis. And now you'll start seeing the beauty coming out with which natural product we started. Now, why did we choose aldose reductase enzyme? It is just random? No, not at all. It is not random that we just took some enzyme and started looking for its inhibition. This aldose reductase enzyme is now being viewed as one of the most important enzymes for its implication to major health problems of the 21st century. And to be precise and very brief, all the diabetes long-term complications which are arising because of the hyperglycemia or the elevated sugar level is partly coming from this activity of this enzyme, because glucose starts getting reduced to sorbitol, and then sorbitol opens many other avenues. In fact, cataract formation in the eyes is now being directly thought to be due to the increased sorbitol production, which this enzyme actually does reduction of glucose, because we have got elevated levels of glucose. Glucose should originally go to glycolytic pathway, that means for energy production, where it gets cleaved to 2 molecules of pyruvic acid and then goes to Krebs cycle and generates energy for us. But glucose also gets open to another pathway called as polyol pathway, where glucose just gets reduced to sorbitol, and then sorbitol, wherever it starts accumulating, starts bringing those complications. So in short, what I'm saying is aldose reductase is a very important enzyme which definitely has a major role to be played, at least in the diabetes in the current stages, but they are looking its role in other areas also. Now, having got rationalized that why we chose this, now we looked for one of the simplest thing where we can look for this good aldose reductase from nature. Now, this Indian gooseberry, which is nothing but your amla or avla, people call it different way at different places. What is the most important ingredient is beta-glucogallin. You can see this, it's nothing but just a ester of 3,4,5-trihydroxybenzoic acid with glucose. That's your beta-glucogallin. What we thought was that we'll make its isoster Why? Because this is hydrolytically unstable. The moment you take this beta-glucogallin, it should get hydrolyzed, and it gets hydrolyzed to 3,4,5-trihydroxybenzoic acid and glucose before it actually go and does the job of aldose reductase inhibition. So we thought that we'll make a very stable analog which doesn't undergo hydrolysis. We envisaged this C analog. Now, why we did this envisage, or why we thought of doing it, I'll come in a moment. That slide is definitely there at the relevant place. This was our proposal. Now, how do you make it? This is the reason why I said synthesis is now coming as a backbone, because all what you envisage, someone has to make the molecules. What we did was, I'm keeping it very simple, we took this 2, 3, 4, and 5 tetrabenzyl gluconolactone and also galactonolactone, depending upon whether you have X as O-benzyl or Y as O-benzyl. When it's Y is O-benzyl, that's your galacto, and when X is O-benzyl, that's your gluco. We added this 2-carbon synthon, that's your nucleophilic addition. We got the corresponding C-connected compound, deoxygenated by the traditional methods, and we got this carbon-carbon bond at the anomeric position in fairly good yield. What do you do with that? Obviously, we removed the ester functionality, hydrolyzed it, converted that to the finerbomide. Now, for the students, it will be nice to see that this carboxy group has to be activated, and this activation was done by pivylyl chloride, where you form the mixed anhydride. In the mixed anhydride, you will have one carbonyl which is statically crowded, the other carbonyl is less crowded, and therefore, when you add this amine, N-methoxy and methylamine, you get the formation of this amide. This is your finerbomide. What's the advantage of finerbomide? That even if you add large equivalents of Grignard reagent, it will stop at the ketone stage and will not get over-reduced. That's the strength of this finerbomide, and we have definitely taken advantage of it. You got this aryl ketone, and now you have to remove the benzyl protections. And what we saw was that we made this compound, this is exactly what we need, 3,4,5-trihydroxy, but debenzylation was a problem. Why it was a problem? What was the problem? Then when we subjected this molecule to hydrogenation, you saw that this carbonyl also got reduced, CH2, CH2. We don't want that. So then we had to do little bit of function protection, deprotection. So benzyl ether protection is removed under hydrogenation. We wanted to avoid hydrogenation at that stage. We removed at the earlier stage. Now you have this, and now we incorporated mom protection. Now you can see on 31, you have mom protection. Mom. This is your methoxy methyl ether protection. Students should get the feel of that these protections can be removed under acidic conditions, because we had problem of removal of these protections under hydrogenation condition. We removed under acidic condition. Obviously, you did the Grignard, you got this ketone, remove under acidic condition, you got your 33, which is your trimethoxy compound. And then just to characterize it further thoroughly, that we have the correct structure, we just peracetylated this in 4 hydroxys here and got this peracetylated product, and you got the compound, this compound actually. And we prepared all what we wanted and obviously subjected them to aldose reductase inhibition studies. Obviously here I need a biologist because I am a synthetic chemist. I just restricted myself to synthesis. So I gave it to a biologist who is extremely competent and had been working on aldose reduction from several years. So we gave it to them, and what the observations or results came were that's only low to moderate activity. We're not very excited about it. But yes, it had some activity, but not great. Now, here is the reason why we went in for this. I promised you that I'll tell you why we went in for this compound. Natural is O. Here you have O. It's an ester. Barbara had tried making it more stable, because this is ester which is less stable hydrolytically. He made the amide X is equal to an H. and observed that that is a very good compound and gave 80% inhibition at 50 micromolar. So it's more stable than ester. And that inspired us to actually making X is equal to CH2, which should be even more stable. And maybe that will be, and this is what we made, this synthetic, which I have written, trimethoxy, and we studied it. It is inhibiting enzyme, but not anything great like the NH1. So that's just the study outcome. But our interest didn't dwindle, and we took it further. Now this plant, licorice, it's known since ages. You go and just type this word in Google, you'll be able to see that this has been centuries and centuries mankind has used it, which is known in Hindi as mulethi, and in English, obviously it has a very sophisticated name. It's Glycyrrhizia, and there are three main varieties of Glycyrrhizia. And what has been observed is that the dried roots and rhizomes of this plant has several natural products of great importance. Saponins, chalcones, flavones, coumarins, and that like that. And it's sweet, and the sweetness is due to this molecule, glycyrrhizin. Now, this plant has also given this simple chalcone. Now, I'm using my word very carefully. Why I say simple chalcone? Because chalcones in the lab can be assembled by just one-step, two-step reaction. And chalcones have been synthesized in the literature since last several decades in the lab. What we observed was that isolicaritin, see the difference between isolicaritin and isolicaritiginin. Isolicaritiginin, if you just add a glucosyl moiety here, that becomes isolicaritin. That means I can confidently say that isolicaritin is nothing but O-glucoside of isolicaritiginin. And this was known to be inhibiting this aldose reductase enzyme. Same plant had also given these molecules. Amorphutin A, you see the difference between them very carefully, amorphutin B. And between A and B, you have the difference. Here you have this unit, whereas here you have that geranyl unit. Now, we were inspired from these two natural products because our intention was aldose reductive inhibition still, and also the PPAR, which I said a moment before at the introductory level. Now, what was our proposal? Our proposal was that if nature has this isolicaritin, and this also is there in nature, and this also inhibits aldose reductive enzyme, we proposed, this is our proposal. See the difference. This is O-glycoside and this is C-glycoside. And why I'm again and again coming to C-glycoside? If someone wants to understand that, O-glycoside, this can be anything, this A-glycone. And the sugar is linked through this oxygen. And they are hydrolytically not stable. They get hydrolyzed. But the moment you connect the A-glycone with through carbon bond, it becomes extremely stable, hydrolytically stable, and definitely have much better results. Many places this has been observed in the chemistry and biology. Therefore, we were inspired to undertake synthesis of these two molecules. What are the difference between these two molecules? Here the double bond, here there's no double bond here. And why we propose this? Because this was davidgenin is also aldose reductase inhibitor. So we thought, okay, solubility will be one good thing because incorporating glucosyl residue will definitely make this more water soluble, which otherwise make these compounds less soluble in water. So again, sorry, we wanted to be very sure this time because just like what happened last time when we took that amla C and log, we didn't get very good result, but we thought, okay, let's do the stalking study. although we can do it ourselves, but we gave it to a colleague who's very competent, and he'll be able to definitely have a greater insight because of his experience. And that's the reason why I'm saying medicinal chemistry is a teamwork. One cannot say that, okay, I have a field of doctoring studies and I can do it and I can claim myself. No, the experts will have different strengths because their insight is definitely greater. So we gave these 3 molecules for docking, and this was done by Dr. Mukhopadhyay using V-Life MDS suite. And what the observations were that isoliquirated genin, it's this airing actually goes inside the enzyme and docks favorably at the right place. And when we take this glucosylated, which we are proposing, it's the other way around. It's the glucosyl ring is actually going inside and docking. So this was really very surprising for us. But sorry. We took this glucosylated pivot genin and also docked it, and we saw that this again has a very interesting thing. It's the same ring like isolated genin which goes inside and docks, and the glucosyl ring is outside. Very contrasting result. But we didn't get deterred by this initial observation. But yes, they were seemingly good molecules. We went on for the synthesis. And here is what my own students just came into picture. How do you make this molecule? Apparently looks very simple. Yes. Please see if we have this aldehyde, which aldehyde 39A or 39B, you have to just do your BSc level chemistry. You have an acetophenone on the side, an aldehyde on the side. You just do your aldol condensation. You should get your product which you desire. But making this building block 39A or 39B was the actual stuff. And I really mean it's an actual stuff, although it looks very simple. Here we took advantage of observation made by Paul Norchel that you can make grignards of aryl iodides even containing electrophilic centers like ester here in 40. By this method, you just exchange the iodine by magnesium bromide. This is done very conveniently at -15. Treat the iodo compound with isopropyl magnesium bromide. Exchange occurs, and then you get the grignard formed here. You want your MGBR to be here, which is going to add to your tetrabenzyl gluconolactone, which I showed you in some slides before. It added, and you got this product. Now you can easily see you have added a grignard onto the anomeric carbon with ester functionality. So this was deoxygenated, usual method, triethylsilane or triisopropylsilane, BF3 ethyrate, and we got this 42 in good yield. And what do you do with that? Obviously, you can convert this ester to aldehyde directly by diabal, but when the student went and did the direct reduction of ester to aldehyde using diabal, he saw that there is always an over-reduction. I think that is the reason why he reduced ester completely with LAH, got this alcohol 43, and then oxidized it with PCC to this aldehyde. And now you can easily see that he has got 39A with benzyl ether protection. And I have already told you how he got the MOM. MOM was coming from this route. You can easily see you remove the benzyl ether by hydrogenation, load your MOM protection by MOM chloride, and then do the same chemistry. That means reduce the ester with LH and reoxidize, but this time he has reoxidized with IBX. And I think it makes sense because here he did oxidation with PCC because he knew that it's going to be a slightly acidic condition, and maybe that PCC is going to affect those MOM. So he has been very careful that he has taken IBX for the oxidation, and he got both 39A and 39B in good yield. And that's what I said. Now you take your acetophenone and do your BSc simple chemistry. What is that? You have acetophenone, you have aldehyde, either 39A or 39B, you get your aldol condensation, and you have got your 46A and 47A. What's the difference between them? Here you have benzyl ether protection, here you have MOM protection, and now you're going to remove the protections. And I'm not going to the details of that because I want to complete my lecture in time. Obviously, you made all of them very elegantly and finally submitted 16 compounds for evaluation at National Institute of Nutrition, Hyderabad, where again, Dr. Bhanu Prakash Reddy is very eminent in this field of aldol reductase inhibition. He did this job for us and found out that some of the compounds are showing extremely good activity, almost 100%, much better than even. So whatever we were demoralized with amla, now it looks like this isolicylregenin gave us good inspiration that yes, we can definitely get good compounds, and they were water soluble. This is the conclusion of this part. What is that? You made this molecule, and I said most of the compounds were water soluble. That's encouraging and very encouraging biological studies. Here are some of the compounds that showed extremely good activity. And again, we acknowledge the efforts of Dr. Bhanu Prakash Reddy, who did this biology. Now I'm coming to the next quick conclusion. I should not say conclusion, but this part, we are extremely excited that in the context that we have been so happy with these results. These results have been just declared in the literature some time back that this molecule is going to be a very useful for neural protection. You know, we have glucosyl residue here, which is water soluble. I'm sure someone can take this work ahead and see its role on neural protection. We definitely would look forward for any biologist to pick up this and show that it can be applied. Now I'm coming to the other natural product, which we said, amorphotine. Again, please concentrate on this point. This is a single bond here, and there's another double bond here. This is from a different plant, Kajani stilbene. This is from that licorice. Both are also occurring in the same plant at the same time. Very interesting. And we thought that if we can make a library of this compound, it will be very valuable for PPAR agonist activity. How do you do it? Again, I'm coming to synthesis. That's our strength. We made this building block, number one. And who made it? This is Dr. Mukhamala. He made this building block. I'll show you in a moment how we made it. You can easily see if we alkylate this position and desulfonylate, you will get left-hand side compounds. And if you do a Julio olefination at this methylene, we will get the right-hand side compound. That's the double bonded compound. And once we have this building block, you should be able to get the left side compound and the right side compound. But they are nothing but they are analogs what we have proposed for amorphotine and Kajani stilbene. And he was successful in making this building block. Now, I'll just quickly go through it because we have paucity of time. How do you make this building block? One, it starts from this natural carboxylic acid, ursolinic acid, but it's expensive. Therefore, the student was encouraged that why don't you make this ursolinic acid in lab, which is nothing but your A written here, A. And this is where he actually contributed very nicely. He optimized this procedure and we could get several grams of A, which is nothing but ursolinic acid. It's expensive, but he made it. And this is nothing but condensation between crotonate and beta ketoester, and then later oxidation by iodine. You get this aromatic compound A. And then you can, it's a usual chemistry, you have to somehow bring bromine, benzylic bromination. And he did, taking A, protect the cycloxy group, brominate the benzylic position by NBS, gets this, but there is a side product problem is also there. And then this benzylic bromide was reacted to 2-mercaptobenzothiazole to get the sulfide, and this sulfide and oxidation should give you your building block 1. Assuming that you have got your building block 1 now, You have to just do condensation. That's what I said. React with aldehyde. That's your Julia reaction. All those who are synthetic organic chemistry students will realize that Julia reaction is nothing but sulfone reacting with an aldehyde, and you directly get your double bonded. We have not gone through the Wittig approach. There are advantages of Julia olefinication, and you got this alkenes. And then what do? You remove your ester functionality or acetyl group. You get, we got large number of compounds. I'm just going through that. You see the number of products he made, all of them in very good use. Obviously, very, very happy to see that he could make library of these compounds. Where, what is the library of this compound? We are changing the substituents in this ring. You can see, we know that this ring is extremely important. We can't touch that. But we wanted to see the variation in this part. And that's the place where your medicinal chemistry comes into the picture, because we had consulted with some of the biologists who were giving us directions. So we made this large number of compounds. And here is one example where we had this pyrone, but we have to be very careful removing this acetyl group because it decomposes. And therefore, he developed a milder method to remove this acetyl group and got this molecule. Okay, so he made that double bonded. How about the single bonded compounds? Take the same building block and alkylate this position. Methylene, alkylate, sodium hydride, and then desulfonylate. So you will get your desulfonylation with samarium iodide, and you get your bibenzyl compound. This is bibenzyl. Please see that there's no double bond here, it's a single bond. Otherwise, you will feel as if it's being repetitive if your attention is a little low at this juncture because of the lunchtime. So you will have this single bonded compound, and he again made several of them, and I'll not go into that. Obviously, we were so happy with this building block approach for synthesis of both the class of compounds. We did patent it. And what to do with this molecules? Obviously, we approached a biologist at this Max Planck Institute for genetics at Berlin, Germany, and Dr. Sassasar was the one who was involved in this PPAR agonist activity, and we were extremely happy to take these compounds and investigate. And to our good surprise, we found that one of the compounds was found to be extremely good, even better than the starting point where we started from. And obviously, we could not follow it further because the scientists from there moved to some other place, but we are still keeping hopes that we will take it further from here. And this was published at that moment because the student was very curious to have publication for his own career point of view. Okay, now this is the conclusion for this part. Where is that? That you have double bonded and single bonded compounds, and they are very good PPAR agonist activity. I'm coming to the last, that is your SGLT inhibitor. And quickly I'll go through because this is already there in the news. Foxiga or Foxiga, these were the drugs discovered a few years back, where it reduces the sugar levels in the blood by allowing sugar to go through urine. How it happens, I'll tell you in a moment. This drug, dapagliflozin, is a synthetic drug. And it's a C-glycoside. That's what I said. Here you have your glucose linked to this A-glycone part. A-glycone part. And this was the first drug in the market which was for diabetes operating through SGLT2 inhibition. What are these SGLT? This is sodium glucose linked transporters, and where are they? And how they allow this excess sugar to pass through urine. Now, to understand that, we'll have to go quickly to a little bit of biology. See what happens, blood comes to the kidney, gets filtered, and purified blood comes out of it. Now, when this filtration is occurring, kidney has been designed by nature that it should reabsorb all of glucose. That's the normal process. But the scientific community thought that, okay, if we can allow kidney to throw excess glucose outside, the load on the body will be reduced. But was it imagination of a scientist? No. There was an observation in the literature, way back, very, very old literature, Now here I am showing a excretion of glucose into urine is referred to as glycosuria. Someone is urinating and most of the glucose is passing out. And there was an observation in the literature, I'll come to that in a moment, but how this happens, how excess can be allowed in the urine. I have just shown you quickly the kidney, same impure blood comes, purified blood goes, filtration is occurring inside the kidney. But who is doing it? Each cell of the kidney, if I see, it's called a nephron. This is one nephron which I have drawn here. And there are millions and millions of nephrons doing the job. And somewhere some filtration is occurring. I can't see there, someone will say. This is the place where the filtration is occurring. Again, blood is coming inside, blood is going outside, but something is getting filtered here. And the filtered liquid is being dropped here. Can I see it? Yes, I can see it in this picture. Here is the filtration occurring. The liquid falls here, and this liquid has to travel a long distance, and then that's the urine coming here. But why? This long path is there so that valuable nutrients can be reabsorbed. And this epithelial cell lining which I have shown here, they have those transporters whose job is to reabsorb glucose from the liquid which is passing through this duct and give it back to the body. But that's what the scientific community thought, that can we inhibit this job? Let the transporters do not reabsorb this, and we'll be able to allow excess glucose to go off through the urine and reduce the load on the individual. And I said, they thought by imagination. No, there was an observation. What was the observation? See, this observation is 200 years old. Floridine was a molecule from the bark of an apple tree, which ingested on gram scale. They saw that most of the glucose was passing through urine. Humans had already seen it. But how it was occurring, no one knew. Obviously, with the advent of time, and biologists started investigating how it happens, why it happens, and they finally figured out that it is because of that kidneys reabsorbing and not reabsorbing. So this information of SGLT inhibition came quite later. And I've given you the first, this also you can see it's more than 70 years old, where actually all these studies were being done. So inspired from that, AstraZeneca and I think Bristol Myers Squibb came up with this molecule, dapagliflozin. You can see here, right hand side. What we thought was, can we make this molecule? Because nature had provided through oxygen link, we should make through CH2 link. And this was the challenging work, which again that same student took up and did the synthesis. Again, the synthesis is fairly simple. If you make this building block and do the chemistry with this, you'll be able to get them. And I'm not going to go to the same thing. Here is that Notchell who made Grignard reagents in presence of electrophilic centers. That inspired us, and the student took real advantage of this observation. And finally, I'm cutting the story short because time is up. Now, here are the final molecules. You can see we made all these molecules, and here is the second edition of the ring coming through andromite functionality. And finally, you'll see we started getting what was the original objective? Original objective was to make the dapagliflozin analog where it is linked through CH2. This is linked through CH2. This is linked through CH2. This is linked through CH2. So these are the C glycosides which we prepared with additional carbon. And finally, all these molecules were made. And here I have to definitely give full credit to our colleague who is now probably sitting there at NIPER, Guwahati, Dr. Sanjay Banerjee, who was earlier in Delhi. We gave all this library of compounds, and he was extremely grateful. We are extremely grateful that he did good job for us. And we saw that many of the compounds are very good SGLT inhibitor. This again goes on to repeat the same thing. Medicinal chemistry is highly interdisciplinary, highly multidisciplinary. We have to have teams working together. Otherwise, we won't be able to make much progress. And I'm coming to the almost last few slides. Have we stopped the journey? No. This was the dapagliflozin. And how the further advances being made? People are trying to change the proximate ring and the distal ring to see if we can bring about even more better molecules. And we have made fairly bold initiatives in this direction. We have made a set of molecules where you have this first ring, A ring as isocoumarine ring, and in the second target, we have made A ring as pyridine ring. And obviously, we await some initiatives to be taken in this direction. Okay, so I'm summing it up. Synthesis is the backbone. You must have seen all this library of compounds which are made so difficultly. It requires a very, very good synthetic hand. And I'm extremely grateful to these 4 students, Dr. Mukamala, Dr. Sudarshan, Dr. Tiwari, and Dr. Rajeshwar Reddy, that they contributed a lot for all the synthetic schemes, which looks like very simple on slides, but when they actually work in the lab, how many difficulties they encounter. And that's the reason why I give full credit to them and reiterate that for medicinal chemistry, synthesis will remain the backbone or the founding rock foundation for the advancement. And I finally thank all of you all sitting on the other side of this system. Now in this internet age, we just address to the system, to a machine, and someone on the other side is listening to us. We assume that all of you all may be very attentively listening, and we give full credits to that and appreciate the patience which you have shown, because given the fact that it's lunchtime, I'm sure everyone is waiting for lunch break. Thank you. Now, last slide. This is, I'll excuse myself. This is a small advertisement. We have been doing medicinal chemistry conference since 2009. You can see we are in the 7th edition now. Every 2 years we do it. And this medicinal chemistry conference is in December 1st week. And we request the student community particularly to register themselves at this site and enjoy this. We have great speakers in that conference, few from abroad also, and also from within the country. I am just keeping the excitement down because we have really good people for this conference being virtual. Thank you. And Dr. Kaplishwar, Dr. Sethi, I am extremely sorry if I have over reached my time beyond the allocated.
No, sir, it's absolutely okay. And thank you very much for your wonderful and insightful talk. Now the session is open for question and answer sections. If you have any questions, please feel free to post in the YouTube link, and from there I can convey the question to our speaker. So if there is any question from participant side, please feel free to write down in the chat box of YouTube link, so that we can convey the question to the speaker.
I think, Kapil, most of them are all set to go for lunch. It looks like that. I hope you are able to see me.
Yes, sir. You are very much visible, and you are audible also, sir.
Good. Because we have to keep asking this all the time, because now in this internet age, these are the two important things: whether the video is being sent properly or not, whether the slides are being sent properly or not, whether people are able to hear. That becomes extremely crucial.
Yeah, I agree fully with you, sir. So, dear students, dear participants, if you have any question, please post into the chat box of YouTube link. So, I think there is no question, and if there is no more question, please join me to thank Professor Singh for his wonderful lecture. We are pretty much honored that you have accepted our invitation, and a very big applause for Professor IP Singh, sir. Thank you very much, sir.
Thank you, Kapil, for once again for the invitation and being so patient with this 50 minutes of talk.
Sir, just now we have got a question from one of the participants. So the question is how the in vivo bioavailability changes going from from oxygen to C glycoside, oxygen to carbon glycoside.
How the in vivo?
How the in vivo bioavailability changes going from oxygen to carbon glycoside.
Yeah, honestly speaking, I won't pretend I don't know the answer for this. Biologists will be able to tell it better than me, those who study. But we know for sure, our objective is only to see that hydrolytic stability, okay, which oxygen doesn't have, whereas the CC bonded compounds have hydrolytic stability. Our objective was only from that point of view. And the bioavailability point of view, I won't be able to comment. I'll definitely invoke a good biologist to speak on that front. No pretension.
So, dear participants, if you have any more questions, please feel free to email Professor Singh. So, he will be more than happy to answer your questions. And thank you, sir. Thank you again for your nice talk, and a big applause for Professor IP Singh, sir.
Thank you.
Thank you, sir.
Yeah, I can leave now?
Yeah. Now, dear participants, please note that it is now lunch break and we'll resume session at 2:40 p.m. So we'll start Sir, shall we start?
Yes, I am ready.
Yeah. So very good afternoon to all of you, dear participants. Welcome back after post lunch session. I hope you all have delicious lunch. So please do not go to sleep mode. And it is my pleasure to introduce next speaker, Professor Prasad B. Bharatam. So Professor Bharatam did PhD. Professor Bharatam is the head of the Department of Medicinal Chemistry, NIPER SS Nagar. He did his PhD from University of Hyderabad on applied theoretical chemistry and subsequently performed postdoctoral research at University of Alabama, Birmingham, USA. In 2001, he joined NIPER SS Nagar, and since then, his research group is engaged in organic synthesis and computer-aided drug design program. Professor Bharatam has published in several high-impact peer-reviewed journals such as Chemical Communication, Journal of Organic Chemistry, Organic Letters, Journal of Medicinal Chemistry, Angewandte Chemie International Edition, Journal of American Chemical Society, etc., with a current H index of 37. He is the recipient of several prestigious awards, Fellowship of Indian Academy of Sciences in 2020, OPPI Scientist Award in Medicinal Chemistry 2009, Ranbaxy Research Award in Pharmaceutical Sciences in 2008, Chemical Research Society of India Medal in 2008, IBM Faculty Award in 2007, to name a few. With this brief introduction, I would like to invite Professor Bharatam to deliver the lecture. Sir, please.
Okay, good afternoon, everyone. It is nice to meet on this platform, which is being organized by NIPER Guwahati. Medicinal Chemistry Department is organizing it. I am happy that this event has progressed very well. Initial lecture by Professor Mehta and then many top class lectures. So I am going to give a lecture on how to design anti-malarial compounds. This is my topic. This is using computer-aided methods as well as synthesis. So the idea behind this lecture is how to take the results from computer lab to organic lab, from there to in vitro biology lab, from there to in vivo biology lab. Whole entire domain I would like to. Translational research is important in the coming many years to come. So this is one small example in that direction which we have contributed in the recent past. So I am trying to share my screen. I hope you can see my. Okay, please confirm. Is it visible, my PowerPoint?
Yes, sir. It is absolutely visible.
Okay.
So the title is Designing Anti-Malarial Agents Using Computer-Aided Methods, Synthesis and Biological Evaluation. Now, very first thing we have to look at is the 3D structure. 3D structure of the target. It is very, very crucial. We do not have the habit of the thinking in 3 dimensions. I wish all the computer-aided drug discovery people should only think in 3 dimensions. If anyone is thinking in 1 dimension or 2 dimensions, that will not help. We have to think in 3 dimensions, everything. For that, computer software is very much helpful. When software is available today, why don't we use that to think in 3 dimensions? Everything in drug discovery, we should think only in 3 dimensions. We should not consider 2 dimensions at all. Forget about 1 dimension altogether. Okay, so what is meant by 1 dimension? Paracetamol is 1 dimension. Molecular formula of paracetamol is 1 dimension. They are not going to help. 2D structure of paracetamol is good. It helps little more than the small thing, 1 dimension. However, 2 dimension is also not sufficient enough. 3 dimension is reality. Drug is in 3 dimensions. Macromolecule is in 3 dimensions. Interactions are in 3 dimensions. Signal transduction is in 3 dimensions. The secondary messenger generation is in 3 dimensions. Everything is happening in 3 dimensions. So we should not be limiting our thinking process to the technology that is paper and pen. When we write on a paper with a pen, we always think in 2 dimensions. But these nowadays, these facilities are available. Computer-aided facilities are available, which are most of them, 50% of them are free, 50% of them are costly. Let us adopt the free ones. Think only in 3 dimensions. That is what exactly we do in our lab. Look at the macromolecular structure in 3 dimensions. That is the first step. Once we start thinking, we can go to the next step, that is structure-based drug design. Structure here means 3D structure of the macromolecule. We do use the 3D structure extensively because we rotate, zoom, represent it in cartoon model, we represent it in ball and stick model, we represent it in quaternary structure model. Likewise, we can easily convert one way of looking at to another way of looking at using the software. That facility is available, and many such facilities are free of cost also. PyMol is a software which is free of cost. Why can't we use it? Mercury is not so very costly. If you buy data bank, it comes with it. In fact, for 1 or 2 molecules, if you want to see, for a limited number of molecules we want to analyze, Mercury software is free, which is coming from Cambridge Structural Data Center. So we have to think in 3 dimensions all the time. In fact, BPharm first year or BSc first year student should be introduced to this process called 3D thinking. Okay, next, pharmacophore mapping. This is another technology coming from computer-aided drug design. QSAR, most of you are aware of this technology, which is based on statistics, quantitative structural activity relationship. Okay, if we can do that, we get lots of insights. Then molecular dynamics. Every molecule is always under dynamical state. Induced if it happens when the small molecule interacts with the macromolecule. That all requires 3D thinking. Quantum medicinal chemistry, that helps us thinking into the molecule, thinking inside the molecule. What are the molecular orbitals? What are the energies? What are interactions? What is the HOMO-LUMO gap? What is the nucleophilicity? What is electrophilicity? What is chemical potential? There are many properties one can estimate with the help of quantum medicinal chemistry, and that we should explore. That also requires thinking in 3 dimensions only. Apart from that, there are surface properties, electrostatic surface properties of the drug, surface properties of the macromolecule. Both of them have to recognize each other. If macromolecule has to recognize small molecule, and if the small molecule has to recognize the macromolecule, there is a need for the surface properties. We can easily estimate them using quantum medicinal chemistry. Maybe little time is required for the computer. We'll give the time to the computer to give the result, but it gives you very, very reliable result. All the computer-aided methods are very reliable results they provide. But drug discovery is an art. Science provides you many facts. But when you go to the interpretation, you have got to learn the art of drug discovery. I'm sure you have some practice, and I'm sure you are going to learn it a lot more in future. Once we design the compound, we take it to the organic synthesis, and from there we go to in vitro laboratory, from there we go to the in vivo laboratory. Then we can complete all the preclinical pharmacological studies. For all that, what is the very first step? It is the homework. If your homework is done satisfactorily using the 3D structures of the molecules, we are done. We are almost ready for taking up to the big level. Now, what is next is we start thinking at very, very small molecules. This is, look at these small molecules, imidamide, that's a small molecule, guanidine, urea, thiourea, selenourea. These compounds are so small to understand the internal details. We spent some time and we published a few papers as shown in the same slide. But we slightly extended this work to bigonides. Bigonides contain 2 guanidine units. They are fused to each other. They are not ring fusing. It is just two condensed, 2 guanidine units condensed to each other. You will get the bigonides. There are so many drugs with bigonide moiety, metformin, fenformin, chlorhexidine, like that. But look at the structures. The structures look very simple, very nice, in very, very simpleton type. Okay, novice structures. But let me tell you, these are all wrong structures. These are all the wrong structures of the bigonides. This is how every medicinal chemist is writing, experimental medicinal chemistry, even the crystallographers. Experimental biological crystallographers are using this kind of a structure for the drug molecules, but this is wrong structure. I'm going to explain that today. That is one of the objectives of today's lecture. Look at the simplest structure of bigonide. Bigonide is written like this in this green color circle. This is how people have been writing. But this is not true structure. This is not the most stable structure of the bigonides. This representation is a misleading representation. Entire medicinal chemistry community is getting misled by themselves by writing it in the wrong way. Suppose I find one of our friends 2 times in the mess, a hostel mess. I think that this fellow is always found in hostel mess. He eats too much. Suppose I make that kind of interpretation, it is absolutely wrong. But the very first impression is like that. So the structure, the position of the hydrogen atoms have got to be appropriately written. The structure 2 is correct structure, whereas structure 1 is wrong structure. That is wrong tautomeric state. The wrong tautomeric state, when we consider, we get wrong impression and we use the wrong thing for many, many years to come. Even in the year 2017, 2019, we have seen research publications of top class journals also using the wrong structure of the bigonides. Well, I'm going to tell you why it is wrong structure. Because it is wrong tautomeric state. The right tautomeric state is here in the structure number 2. What is the right about it? This structure 2 has got conjugation. Structure 2 has got a stronger hydrogen bond. Structure 2 does not have protonation, the proton at the N4 center. That means there is no pyramidalization in this structure. Because there is no pyramidalization, there is the hydrogen bond is strong. If the hydrogen bond is strong in structure 2, then it is more stable structure. There is no allylic strain in structure 2, but whereas in structure 1 there is allylic strain. And then there is protonation. Protonation is happening on structure 2. If protonation happens at structure 2 at the N6 position, this structure 3 is observed. Protonation does not happen at N4. Cannot happen at N4 because protonation at N5 and 6 center is really the most stable and more preferred structure. Then you look at the arrow given here. Because structure 2 is right, it can also show 1,5 hydrogen shift. Structure 2 is having the resonance stability due to hydrogen bond assisted delocalization of the electron density. This is also known as hydrogen bond assisted aromaticity. So in this background, you can see that structure 3 is structure 2 is quite reliable, whereas structure 1 is indeed wrong structure. So if it is wrong structure, why don't we try to make a correction on this? This is the energy difference. The right structure versus the wrong structure, energy difference is 9 kilocalories per mole. If you go to the Boltzmann distribution, between the structure 1 and structure 2, N2 by N1 or BG1 by BG5, the number of particles of BG1 versus number of particles of BG5 is 1:2 million. That means if one molecule of wrong structure is present, there are million molecules of the right structure is present in the reaction mixture. So we should completely ignore the wrong structure because of 9.34 kilocalories high energy difference. And then we should go to the protonated form and go to the N-inversion barrier. The hydrogen shift, 1,5 hydrogen shift, that is sigmatropic rearrangement, requires only 2.7 kilocalories per mole. That means at room temperature, if you take any bigonide, it is always undergoing 1,5 hydrogen shift. That we cannot ignore. These details you can get when you look into the molecules with the help of quantum medicinal chemistry. Now, look at the surface of the molecule. Look on the surface. Look at BG1. BG1 is from the right tautomeric state. BG5 is the wrong tautomeric state. Look at the surface of the molecule. Surface of BG1 is quite relevant, whereas structure of surface of BG5 is not relevant at all. Okay, the surface of BG1 is complementary to that of the macromolecular surface. That means the binding between BG1 and the bigonide, that is D, figure number D in this figure, in this whole chart, you can see that there is complementarity between BG1 and bigonide complementary surface. But look at BG3. BG3 is a tautomer, which is not correct. BG5 is wrong tautomer. So both BG3 and BG5, they do not possess the relevant surface property. It is very similar to that. If you are walking on the street and if you see a classmate of yours, if you smile at the other fellow, you will understand, okay, you will understand something. If you don't smile, you simply ignore that fellow who was your classmate last year. Today you ignore him. So he will understand something else about you. If you smile at him, or if you wave at him, if you greet him, he will understand you. Okay, this boy, this fellow is still is my friend. If you simply ignore him, he will say that chaddo yaar, mujhe kya padi hai? I don't have any interest in this fellow. So you will simply do your job, or the other fellow will do his job. But maybe what goes away if you make a smile? That is the state of the mind. If the state of the human mind reflects on the surface of the face, on the face of the human, exactly like that, the state of the drug action of the drug action state of our drug molecule is appearing on the surface of it. That is BG1. The right tautomeric state has got the right surface property. In the wrong tautomeric state, BG3 and BG5, it has got wrong wrong surface property, and these BG3 and BG5 will never be recognized by the macromolecule. Only BG1 will be recognized. But let us take it to the little more. Protonated BG1. If you take protonated BG1, it becomes BGP1. BGP1 is complementary to bigonide. This is surface. So BG1 and BGP1, there is not much difference. BGA2, that is the anionic state of the bigonide. That means whether bigonide is neutral or whether it is a cationic state or whether it is anionic state, it has got the right surface property. Whereas when it is in the wrong tautomeric state, it does not have the right surface property. That means right tautomeric definition of the given drug molecule is required to define the right surface property, and that right surface property leads to recognition between small molecule and macromolecule. Because of that, we need to look into the molecule with the help of quantum chemistry, and we look on the surface of the molecule with the help of quantum medicinal chemistry. Both are very, very crucial. I'm sure that the same thing was studied in the year 2005, and we have been able to realize that if we write the structure of proguanil in the right tautomeric state, the pharmacophoric features are quite easily visible. Whereas in the wrong tautomeric state, that is the right hand corner structure on the top, it is a wrong tautomeric state, and wrong tautomeric state leads to wrong message. So we should not be considering the wrong tautomeric state of the drug molecules. Now, similarly, protonated state of bigonides. Drugs like all bigonides, like metformin, chlorhexidine, these are all being used daily. For example, some of you might have used the chlorhexidine. This is nothing but that blue-colored bottle that a dentist hands over to you when you have some problem in your teeth. The dentist says that this costs around 60 rupees. You buy this and rinse your teeth for the coming 1 month every day for about 5 minutes. Then all your teeth problems will go away. So that is nothing but chlorhexidine. That is cationic in nature. That is not neutral. So we need to know whether the drug is acting in the cationic state, in the anionic state, or it is acting in the tautomeric state, or what is the state of the drug. We need to be able to understand that. So all this comes in comparison to a beautiful chemistry called carbon chemistry. There is lots of details. I don't want to give much information about carbones today, but this is about molecular orbitals. You can see the sigma type orbital, pi type of orbital, and then we can say that because of the nitrion type character of bigonides in their cationic state, they are always in cationic state. You do not find bigonide. Nobody will supply neutral bigonide. Sigma Aldrich cannot supply neutral bigonide. They only supply cationic bigonide. That is the HCl salt of the metformin and others. Okay, so because of that, the N inversion becomes easy. It requires only 9 kilocalories per mole. Rotational barrier is easy. It requires only 5.4 kilocalories per mole. Very simple. That means after protonation, bigonide is not only becoming active, it is jumping and dancing because it becomes highly dynamical. N inversion is possible in the protonated state. It is not possible in the neutral state. CN rotation barrier is possible in the dynamic in the protonated state, and it is not possible in the neutral state. In fact, bigonides are not drugs at all. Metformin is not a drug. Metformin hydrochloride is a drug. There is a large difference between metformin and metformin hydrochloride. It is just remember there is a young boy who is looking for a job and he gave a good interview. He is waiting for the appointment order. He is not getting the order. Monday, Tuesday, Thursday, Friday, all these days he is waiting. He is dull. He is looking depressed. He is about to see why the letter is not coming. But suppose he got the letter. And he received the letter telling that from tomorrow onwards you can join our company. Your salary is 1 lakh rupees. See the difference of the performance of the boy. Depressed person, metformin is depressed. Metformin hydrochloride is active, dynamical, dancing and jumping, just like that boy who got the job. So metformin hydrochloride is the only drug. Metformin is not drug. We are misleadingly, we are telling ourselves that metformin is the drug. It is not correct. So we have to remove some of these misconceptions from the minds of all our scientists. Now, similarly, there are other complexes. This is cycloguanil, which is anti-malarial compound. Then silicon compound is also there. Boron compound is also there. They are also tested for biological activities. Now, how to convert from proguanil to cycloguanil? What is the energy of that? What is the mechanism of that? What is the structure of the transition state? How much energy is required for the entire? All these things can be easily estimated, and we can understand how this proguanil to cycloguanil conversion is happening inside the human body. In the year 1942, it was realized proguanil is not in the excreta of the human when it is supplied. It is treating the malaria, but the proguanil is not found in the excreta. Cycloguanil is found. That means proguanil is getting converted into cycloguanil. That data was known in 1942, but how it is happening, that became clear only in the year 2014. Okay, with the help of the computer-aided drug design. There are several other molecules one can design, the coordination bonds, but look at the drugs. Ebrotadine, famotidine, they are H2 receptor antagonists. That means they are antacids. That means they are available in a nearby chemist shop. Just for maybe for 1 rupee, they give you 2 tablets. So they are so easy, cheap, and very effective drugs. People are using them as antacids. Now look at fungicidal compound. Fungus treatment can be easily done with this. And then neuroprotective agent, that is also thiazole-based compound. All of them are carrying this nitrione character. What is nitrione character? How to synthesize them? This has become a hot topic after 2010. So there are many methods of synthesis and many details about the molecular orbitals that is all obtained. Look at this synthetic route. Number 1 and 2 can be combined to give you number 3 and 4. Number 3 and 4 can be combined to give number 5, which is a nitrione. So this is one way of synthesizing the nitriones. The other way of synthesizing is also given, but it has to be trapped because it is not really available compound. But then can we have understood lots of studies using computer-aided drug design. Very nice. That is good news. That is all theory. If it is all theory, can I have a non-theory result from here? Can I use this information to design and synthesize better drugs? This is the question. That question can be answered from this slide. A series of guanyl thiourea compounds, that is one side thiourea unit, another side it is guanidine unit. When these two are combined, guanyl thiourea derivatives can be obtained. They are all S-alkylated species. This guanyl thiourea unit is a head group. Such a head group helps in binding with the Plasmodium falciparum dihydrofolate reductase, that is PFDHFR. If these compounds are binding to PFDHFR, that means anti-malarial effect can be realized. If anti-malarial effect can be realized, why don't we synthesize? First do design, then do docking, then you do pharmacophore mapping, then you do molecular dynamics. After doing all that, you confirm, yes, this class of compounds are stable, this class of compounds have got the same nitrion-like character, and thus these compounds are very, very useful for drug discovery. After that, you synthesize these compounds. How to synthesize? We don't have to invent new organic chemistry here. This is not the purpose of a medicinal chemist. The purpose of medicinal chemist is to generate novel molecules which will exhibit therapeutic action. So in this background, we can easily synthesize using the known simpler chemistry. We can condense molecules, we can eliminate HBr or HCl, and using some simple chemistry, we can synthesize molecules like this, and about 50 compounds were synthesized. Out of that, it was found that about 23 compounds are showing therapeutic action. Out of 23 are showing what action? In vitro action, not therapeutic action. In vitro action is also can be interpreted in the form of therapeutic action, no doubt. But inhibitor action is different and therapeutic action is different. If an enzyme is inhibited, that means it is inhibited. That does not guarantee that it will show the drug-like character. So efficacy affinity may be very high, and inhibitory potential may be very high. But whether it is efficacious or not, we cannot answer from the simple inhibitory studies. Anyway, we are not interested in that right now. We are interested to know, out of those 23 compounds, is there anyone which is able to show the therapeutic action? They are all organic molecules. They may not all be drugs. If every organic molecule becomes drugs, it is okay. We don't need so much work to do. But there is a lot of gap between the drug and organic molecules. So that is what we need to understand. Look at molecular orbitals again. Look at metal complexes and synthesize more compounds. And then you go to nitrion type compounds. Obtain the crystal structure and analyze their physical chemistry, electronic density chemistry, and many other things. Look at their molecular orbitals and design more molecules. You can keep on doing this cycle and then synthesize more molecules. These compounds are called azenes, and these azenes are very important drugs, especially one compound called, this is antihypertensive compound called guanabenge. Guanabenge is an azene derivative. This is wherever there is C double bond N, single bond N, say double bond C. When such a functional group is present, it is called a gene compound. And that is that similar many compounds were synthesized. After having done that, one can do this, look at the docking results. What is this docking result is telling? The docking result is giving two messages. Message one is that the lead compound is occupying the same site as that of dihydrofolate. So that is good news. That means the compound that I am synthesizing is showing better affinity towards the Plasmodium falciparum dihydrofolate reductase, and thus it is able to inhibit the drug action. Very nice. But what is the next message? The next message is that NADPH is not being utilized when an inhibitor is bound. When there is no inhibitor, or when there is wrong type of inhibitor, when the inhibition is not successful, then the chances are very high that this compound will become an inhibitor as indicated by our docking results. So what is the biological relevance of this docking? If NADPH is utilized, because this particular pose prevents the utilization of the NADPH. If NADPH is utilized, the drug is inactive. If NADPH is not utilized, then the drug is active. This is the biological relevance. This is the in vitro relevance. We get the signature required for in vitro experiment from the computational experiment. That is exactly the message from this work. Later on, many compounds were designed. Molecular dynamics was carried out. After doing molecular dynamics, it was realized that the designed compounds, they are showing very good, strong binding affinity with wild type PFDHFR, quadruple mutant PFDHFR, double mutant PFDHFR, as well as another double mutant PFDHFR. That means the molecules that are designed, they are efficacious to help in drug resistance also. Not only that they are therapeutically important, they prevent drug resistance. Because with the human DHFR, the binding affinity is only 22. But with PFDHFR, the binding affinity is very high. That means binding affinity with PFDHFR is competitive. That means if we use a particular dose of the drug, it will show action only on the Plasmodium falciparum. It will not show action on the human DHFR. That means human is safe, whereas we are treating the malaria. This is the message coming out of this molecular dynamics analysis. What is the origin of this confidence? It is because aspartic acid 54, it is because arginine, 122 of the enzyme and arginine 70 of the human. You see, with arginine 70, it is showing destabilizing effect. That is a black color. With all other Plasmodium Plasmodium falciparum, it is very strong binding, and with human it is negatively binding. That means that particular amino acid is responsible for the selective inhibition. So we are able to get that kind of information from the molecular dynamics. After that, we need to go to the in vitro laboratory. What is in vitro laboratory telling? What is the message coming from our docking? Our docking is telling, do not worry about any other signature. Pay attention to only one signature. What is that single signature? That is whether NADPH is utilized or not utilized. If NADPH is utilized, that means the molecule is not showing inhibition. If NADPH is not yet utilized, that means the molecule that I have designed, The molecule designed using computer-aided methods is inhibiting the enzyme. That means there is an inverse relation. If an ADPH is utilized, there is no inhibition. If an ADPH is not utilized in your biochemical experiment, that means the inhibition is taking place. So exactly that is the biology that was done. Very simple biology. Simple docking leads to simple biology. If your homework is thorough, you will get clues for the chemistry person, for the biology person, for the pharmacology person, for the safety pharmacology person, for the preclinical drug delivery, preclinical formulation person. Then it is also for the toxicology person. It gives you many, many clues. Computer-aided drug design gives us lots of clues. Sometimes they are mesmerizing number of clues. It is our job to find out what to pick up, what not to pick up, which signature is relevant, which message coming out of the computer-aided method is relevant for my in vitro experiment, which is relevant for my in vivo experiment, which is relevant for the therapeutic action, which is relevant for the selectivity. These are the points we have to mind. Computer always gives you some result. Some result is not the real result. Okay, we should not be trusting computer-aided drug discovery blindly. We have to use our chemical intuition. We have to use our biological intuition. We have to remember all the factors of biology. That means we have to do homework. Before doing computer-aided design, we have to do lots of homework with the help of chemistry, with the help of biology, with the help of pharmacology, with the help of pharmaceutics, and every branch of science. Then we should interpret our computer-aided drug result. I am sure the result will be successful. We do get the signature, but signature does not come automatically from just by looking at the docking results or by looking at the dynamics results. So that is exactly what was done. We paid attention to lots of biology, and then again we paid attention to lots of pharmacology. We have taken these compounds. About 100 mice were used. They were injected with Plasmodium berghei. That means these mice became malarial mice. They became malarial patients. Then we supply our inhibitors. Our inhibitors, which are promising inhibitors, they were supplied. And then as a result, the health of the mice was observed. After 7 days, some of the mice, many mice are okay. But after 14 days, several mice died. But in the case of 5 compounds, that means mice to whom these 5 compounds were injected, actually not 5, 6 compounds. 6 compounds were those mice for which 6 compounds were injected, they have eliminated malaria from the mice. That means the inhibitor is doing the inhibitory action in the mice. Okay, so we are happy. Out of 23 compounds, 6 compounds turned out to be promising. Then we observe the result up to day 60. Observe first 4 days, then 7 days, then 14 days. Daily you observe them. Keep on observing them till the day 60. It turned out that in the case of 5 compounds, malaria recurrence was noticed. Malaria recurrence means those compounds are the compounds which do not treat malaria, but they inhibit PFDHFR in the mice. If they are inhibiting in the mice, that is not sufficient. They have to cure. The malaria in the mice should be removed and permanently removed. So in the case of 5 compounds, malaria recurrence has happened after day 14 and before day 60. So it was realized that these 5 compounds are useless in practice. But in the case of 1 compound, malaria recurrence did not happen for 60 days. All those mice which were treated with this one single compound, they are cured. They have lived their natural course of the life. That means that we have got the message that this one single compound is curative. That is what we want. Now that means if you find one compound out of 23 is curative, that means your homework is thorough. Alternatively, if your homework using computer-aided methods is thorough, you will never fail in your experiment. You will get some positive result. With the desired positive result you will get. So this is the message. These are the 2 compounds which structures are also given. ICVT, in vitro ICVT values are given. Some results are already published, and then I invite you to read these articles which are on the same topic. And now we are going for the patent of this class of compounds. Now, what is the message coming from this lecture? The message is that we have to do homework thoroughly using computer-aided methods. It is cheap to do the homework. Cheap means cheap in terms of finances, cheap in terms of time, cheap in terms of the facilities that need to be created, cheap in terms of the total time taken by the student to perform the work. Okay, so in this effort, the total amount of effort, total amount of work done was about 4 months using computer-aided methods, and then 6 months using the in vitro and in vivo methods. And within 1 year, we know that how to find anti-malarial drugs. Okay, why? It is because homework is thorough. Homework is done at the tautomerism level. Homework was done at the electronic level. Homework was done at the surface level. Homework was done at the synthesis level. Homework was done at in vitro level, in vivo level, and safety pharmacology level, and many other things. So certainly this kind of approach is going to give us very good results, quick results, because we are going to fail early. In drug discovery field, people always give this reminder to each other: fail early, fail better. Failing is nothing wrong in drug discovery research. But assuming that you have succeeded is absolutely wrong. If you don't fail early, you will invest lots of money, lots of time. Maybe before clinical research, already you may spend 3.5 years. 3.5 years on a wrong molecule, on a wrong set of electronic parameters. 3.5 years becomes a very, very big waste. Okay, so before you spend billions of dollars, you perform the computer-aided drug design, which requires a maximum of about maybe 20 lakhs. I'm telling maximum of amount 20 lakhs. It may be sometimes only 20,000 rupees. Okay, so it is possible to do good homework with the help of computer-aided methods, and then new chemical class of compounds were identified, a series of nitrions were identified, and the properties of the nitrions were done, and then chemical bonding was understood, then GTU derivatives were designed, and they were tested in vitro and in vivo, and then you will get many results. And some results you can publish, some results you can patent. So some of them one day it may click, and you may become a very rich man. If you want to become rich, also you should do thorough homework. If you want to become successful, you have to do thorough homework using computer-aided methods and take the results to the organic lab. You should have the guts to take the results from the computational lab to organic lab, and even organic chemist needs to be brave enough to synthesize the computationally designed molecules. We have done lots of synthesis. We have tried to synthesize billions of compounds, which are nature provided to us. Natural compounds, we end up in synthesizing 0.1 milligram of natural product. We say that we have done excellent work, we deserve a Nobel Prize. That kind of organic chemistry was also done. But why don't we synthesize computationally designed compound? It is important. Especially drug discovery people should adopt computer-aided methods thoroughly in the very beginning and do thorough homework, and then only spend time in organic lab, spend time in vitro, in vivo, safety pharmacology, and preclinical formulation. Only after that you take decisions. Okay, this is my message, and I thank all of you and I thank my teachers, and I thank the funding agencies and those people, those agencies which have recognized our work and helped us building our own confidence, and all my PhD students who worked in this area. Those yellow colored names are all the people who directly worked in this area, and all others contributed indirectly for the success of anti-malarial drug discovery in our laboratory. This is a photograph of a few photos of our lab, and you are welcome to visit Naipur Mohali. Of course, you are all welcome to come to Naipur Guwahati also. We are all Naipurs are very progressive, and I want you to take advantage of the presence of Naipurs in India. Okay, thank you.
So, thank you, Professor Bharatam, for your nice lecture. And the session is now open for question and answer. So, dear participant, please feel free to put your question in chat box. So, sir, we have one question. So, the question is, how does the chirality of molecule affect docking score?
It very significantly affects the docking score, because the R isomer versus the S isomer quite often provide different pharmacophoric features. The pharmacophoric features are same, but the order of the pharmacophoric features are different. It is again very similar to the example that I gave you. If you find a student is always found a PhD student in the 5th year, instead of working in the laboratory, if he is found in the sports section, he is organizing sports, he is organizing cultural events, he is not finishing his thesis, what is the message you will get? Okay, you get some message, whatever it may be, whether you think it is positive or not, it is your choice. But a student who is in the laboratory gives you one message, or a student who is in the sports section all the time gives you a different message. Exactly like that. R isomer and S isomer can give completely different pharmacophoric features. Set of features are different. However, in the drug discovery process, it is known that there are many examples where racemic mixture is also therapeutically important. And in some cases, a particular stereoisomer is therapeutically important. So when you are doing docking, if you better dock both R isomer as well as the S isomer, and then correlate your results with the already reported biology, already reported chemistry, and take informed decisions. Which isomer is more efficacious, which isomer is giving better binding affinity, and which needs to be pursued, which needs to be ignored. There are several reasons why we may use only racemic mixture. Then both R and S are okay. Both are okay. But sometimes we need specificity. We have to pay attention to those details. Yes. I hope I am able to answer.
Yes, sir. Thank you, sir. So let me check if there is any other question. Dear participants, please feel free to ask question. Put your question in chat box if you have any. So you can ask Professor Bharatam your questions through email. So Professor Bharatam will be more than happy to give answers through email. So if there is no more question, please join me to thank Professor Bharatam for his wonderful talk. A big applause to Professor Bharatam. Thank you, sir. Thank you very much.
Thank you, Dr. Seth. I am stopping my sharing.
Yes, sir. Thank you. Thank you, sir.
Bye.
So, yeah. So our next speaker is from abroad. So he is sitting in Indianapolis. So it is kind of early morning there. So our next speaker is Dr. Kanhaiya Singh. So how are you, Dr. Kanhaiya? How is the weather there?
It's fine, not raining. Yeah.
Yeah. So let me give a brief introduction of Dr. Kanhaiya. So Dr. Kanhaiya Singh is an assistant professor of surgery at the Indiana Center for Regenerative Medicine and Engineering, Indiana University, Indianapolis. He did his PhD from BHU, India on genetic analysis of impairment of diabetic wound healing. During his postdoctoral research at Ohio State University and Indiana University, he studied epigenetic mechanism of gene expression under diabetic conditions. He has significant experience in high throughput mass spectrometric proteomic analysis, including in vitro and in vivo SILAC. His recent work includes implication of single-cell transcriptomics and single-cell ATAC sequencing approaches to investigate the molecular pathways that are compromised in diabetic wound healing. Dr. Singh has published in several first-tier peer-reviewed journals such as Nature Nanotechnology, Nature Communication, Molecular Therapy, Diabetes, etc. Recently, he has received prestigious Department of Defense Discovery Award from US Defense. And with this brief introduction, I would like to invite Dr. Kanhaiya to deliver the lecture. Dr. Kanhaiya, please.
Thank you, Kapil. Let me share my screen first.
Sure.
So, are you able to see my screen?
Yes, yes. So please share the PPT.
Are you not seeing the PPT right now?
So we can't see the PPT, so we can see the entire screen.
Okay, let me stop share and try again. Are you now able to see the PPT?
Yes, yes. So it is clearly visible. And yeah, please go ahead.
Okay. So, thank you, Kapil, for this introduction. And as you mentioned, I'm working in Indiana University, Indianapolis. And my general interest is epigenetics and tissue regeneration. And since this is a medicinal chemistry conference, I will be talking more on application of genomics in tissue regeneration. So the field of nutrigenomics is a balance between nutrition and how it implicates on certain metabolic symptoms and diseases. So it is a sequential events of nutrients. They effect on epigenome, on the DNA, on the RNA, on the protein. And then if the nutrition is good, it will give rise to a healthy physiological state. But bad nutrition can affect DNA, RNA, and protein, and through metabolites, it can lead to oxidative stress, metabolic stress, inflammation, neurological diseases, and they finally release into certain diseases. And one of the diseases that I'm interested in is diabetic wound healing impairment. It is a secondary complication of diabetes, and I will be talking more about this implications. And I will be talking how it affects epigenome, transcriptome, and proteome of the individuals. So as we know that environmental stimulants affect the disease phenotype through various modulators. Few of them are non-coding RNA, DNA methylation, and histone modification. So like the socio-environment effects that we generally observe, like food, smoking, pregnancy, psychosocial effects, pollution, they all modulate the DNA or RNA through different mechanisms. And the first of that I will be explaining will be microRNAs and their implications in wound healing. So during the wound healing, it is a very sequential, orchestrated events in which there are lots of genes are transcribed. But if all those genes will go and make into protein, there will be a lot of noise. To cut down this noise and give you a specific signal, there are certain regulators. One of such regulators is microRNA. Then they cut down the noise, and then they will give you a meaningful signal. So wound healing is a sequential event, as I mentioned, which start from hemostasis, inflammation, proliferation, and remodeling. In all those steps, microRNAs play an important role. For example, pro-inflammatory microRNA, like microRNA 155, they targets these genes like internucleins and others. MicroRNA 16, they targets inflammatory molecule like COX2, and then they bring down or bring up the inflammatory cascade during the inflammation phase. Similarly, during the proliferation phase, for example, microRNA 21, they can regulate tissue genes like TIMP3, TIAM, or anything, and then they will result into proliferation phase. So most of my talk will be based on this proliferation phase and a specific phenomena called angiogenesis, in which there is formation of new blood vessels to replace or to perfuse the granulation tissue, which is now forming after the wound cascade. So microRNAs can control wound angiogenesis. So during wound healing phase, like for example, wound at day 7, they need proper supply of angiogenic vessels to support the newly formed granulation tissue. And in this, there should be a proper balance between pro-angiogenic microRNAs, for example, microRNA 126, and anti-angiogenic or angiostatic microRNAs like microRNA 200B. So during past decade, our lab is working on microRNA 200B, and we have shown that during the process of wound angiogenesis, in order to set the process of angiogenesis, the microRNA 200B levels should go down. But in certain physiological or pathological conditions, such as diabetes, this do not happen. So our goal is to learn how and why microRNA 200B remains elevated in diabetic individuals, and what are the implications of that? What is the genomics or proteomic effect that microRNA 200B does when it remains elevated in the endothelial elements of diabetic wounds? So as I mentioned, endothelial elements. So it is very challenging to get only endothelial cells or endothelial element, not the other cells. So for that, we are using a technology called laser capture microdissection. So it is a technique in which you have a tissue section and you mark the area that you want to capture, and then ask the laser to cut that area and give that area into your PCR tube, on which you can do certain genomics like RNA-seq or proteomic analysis. So in this example, we have done like CD31. CD31 is a marker of wound and of endothelial cells. So you will mark the CD31 positive element and then ask laser to cut this area, and you will have now a pure endothelial element population, and then you will see the gene expression changes. So here in diabetic individuals, in the wounds, you are seeing the microRNA 200B levels remain high. So what does it does? Like if the microRNA 200B is elevated, what it does to endothelial cells? So it causes endothelial dysfunction. So in a in vitro setting, in microRNA 200B, if we expose to hyperglycemia, 25 millimolar of glucose, we see that the microRNA 200B remains high. And then it leads to endothelial dysfunction. So in this example of endothelial functionality test, we have done matrigel tube formation assay. So if you put any endothelial cells into a matrigel or any other cells also, which has a capacity to form tubes, it will form tubes. But during hyperglycemia, since the physiological conditions are not good for the cells, they are not able to make tubes. That means the endothelial function is lost. But in the same hyperglycemia condition, if you inhibit microRNA 200B, then you can rescue this tube formation. So this supported our hypothesis that elevation of microRNA 200B is not good for endothelial cells in condition of diabetes. So then we asked, okay, microRNA 200B is high. What does it does to the protein levels? So we need to do proteomics for that. And for this, we implicated a technique called SILAC. It's stable isotope labeling of amino acid in cell culture for quantitative proteomics. So I will just explain a bit what is SILAC. So in this, we have a media without essential amino acids. For example, lysine and arginine is not there in the media. And also the FBS that we use is without any amino acids. So that means if we culture the cells and if we provide amino acids from outside, it will only take and metabolize those amino acids on which they are being grown. So then during this labeling procedure, we either provide light amino acids, normal isotopes of carbon and nitrogen, or heavy amino acids like C13 or N15 isotopes of carbon and nitrogen. And then we culture those cells for several passages so that with this passage, all those cells are either labeled by light amino acids or labeled by heavy amino acids. So then during incorporation analysis, you need to see that for a given peptide or a given protein, there should be a mass difference between light and heavy isotopes. And once you see that there is 100% incorporation of these heavy amino acids, then only you can proceed with your actual experiment and mass spectrometry. So for this example, in this, we had cultured light cells in normal glucose media, while the heavy cells in hyperglycemic glucose media. And then we inhibited or inhibited microRNA 200B in that to see what is the effect of microRNA 200B inhibition to endothelial cells. After this experiment, we will mix the cells 1:1, do the protein isolation, do 2D LC-MS to see that for a given peptide, what is the quantitation and what is the value in normal glycemic condition and in hyperglycemic condition. This is very it is very specific, and it gives a very little background noise, because you are running both control and treatment in the same mass spectrometry run. So this is the analysis that we got. So we got around 319 proteins that were upregulated when we inhibited microRNA 200B during hyperglycemia condition. And then we put that into our ingenuity pathway analysis to see that which pathway is affected most by if we inhibit microRNA 200B during hyperglycemia. And we saw that mitochondria, like 2 of them, were coming. That means if we inhibit microRNA 200B in diabetes and in hyperglycemic condition, mitochondria can be rescued. So what was the validation for that? For that, we did an assay called Seahorse assay. It is a real-time measurement of mitochondrial dynamics in living cells. So in this, we used cultured HMECs, human microvascular endothelial cells. And this is the how we measure our mitochondrial functionality. So first, we will let the cell respire in its normal condition to measure the basal respiration. Then, sequentially, after 20 minutes of their basal respiration, we will add oligomycin. Oligomycin is an inhibitor of ATP synthase. It inhibits complex number 5, and hence ATP production goes down. So degree of this down can measure how much ATP is being produced by the cell. Then after the ATP production is measured, we will add FCCP, which is an uncoupling agent, and that disrupt mitochondrial membrane potential. That means now the cell will start to respire at its maximum, and then you will measure the maximal respiration. After this, after the maximal respiration, you will add a combination of 2 compounds, retinone and antimycin. These are complex 1 and complex 3 inhibitors. They will shut down the mitochondrial respiration. And then you can measure, if the mitochondrial respiration is 0, then you can measure what is the non-mitochondrial oxygen consumption. And with this analysis, we are able to found that, yes, in condition in hyperglycemia, if you inhibits microRNA 200B, then your mitochondrial respiration goes up. So this was all the assays that we did for microRNA 200B, and we found that yes, in hyperglycemia condition, if you can inhibit microRNA 200B, then you can rescue the endothelial cell functionality. Now, the next question that we asked, that what are the upstream regulators that is controlling microRNA 200B during diabetes condition? And one of the classical molecule that we got was ZEB1. ZEB1 binds to microRNA 200B promoter, and it acts as a repressor of microRNA 200B. So, and microRNA 200B is known to be an epithelial phenotype modulator, while ZEB1 is known to be mesenchymal phenotype modulator. So then, to see the effect of microRNA 200B, we tried to study ZEB1. For this, we developed a mice called ZEB heterogeneous mice because homogeneous mice is lethal. So first, we validated this mice. So in wounds of day 7 of these mice, in normal wild-type animals, there was a lot of EMT going on. You can see over here, E-cadherin is a epithelial marker, beta-catenin is a mesenchymal marker, and then the co-localization of both of that gives you the EMT score. So over here, you are seeing green and red to be colocalized and more EMT phenomena going on. But in Zeb hetero animals, you are not seeing that much of mesenchymal marker. So that means our animal was good. So using this animal, we first started to look what is the implication of Zeb and microRNA 200B in the implication of wound angiogenesis. So for this, we did an angiogenic blot array, and we found that yes, the certain angiogenic molecules like angine CD26, they were all down compared to wild type in Zeb knockout animal. So then we did the wound perfusion assay. So this assay is called laser spectral imaging, in which you can measure the perfusion of live animal, and you can follow the same wound for different days and see how it is getting perfused after wound angiogenesis. And you can see at day 7, the wild type, there was a good perfusion, but in hetero animals, the perfusion was very less. This was also supported by histological analysis, in which we co-localize CD31 and VWF. Both of them are angiogenic markers, and we can see over here in Zeb animals, the co-localization was very high compared to the wild type animals. So then we proceeded for functional assay, and we do it through Vivo. It is a ultrasound measurement for small animals, and you can see over here the blood vessels over here in a live animal at the wound site, and then you can gate these blood vessels to measure the pulse pressure at the wound site. And over here you can see in wild type animals the pulse was good, but in Zeb animals, as we were calculating, like the perfusion was not good, even the pulse pressure was not good. So what does it means? Like if angiogenesis is less, what is means? That means maybe the wound healing is not complete. So then we try to see the wound healing curve. So if you see the closure of wild type animals and compared to that, the Zeb animals, the wounds remained open. And then it was validated through H&E staining, hematoxylin gyline staining, and KRT14. So KRT14 is the epithelial marker. It can mark the epithelialized tissue, but in Zeb knockout animal, you are not seeing this kind of epithelialization. So, but this was also supported by a functional study. In this study, we measure the wound volume. So now this is a mice wound, a stented mice wound, and you are seeing a wound edge over here. So this can be replicated in ultrasound, and you can see the stents over here, and then the edges of the wound, and this is the wound bed. And with time, you can measure how much this wound edge is migrating towards the wound bed and the wound is closing. So then you can calculate the wound volume by applying some formulas, and you can see over here too that in wild type animals, the wound closed properly, but in Zeb animals, the wound remained high. So with these two implications, like in microRNA 200B and Zeb1, we thought that yes, Zeb1, since it is a repressor of microRNA 200B, in diabetes, its levels should be down because MA200B is up. So we wanted to look what is happening to Zeb1 during hyperglycemia condition. Our hypothesis was Zeb1 should be low, but when we captured CD31 endothelial elements, and we saw that in diabetes, actually the levels were high. So we try to see what is happening in vitro condition in endothelial cells, and here also you can see in hyperglycemia conditions the levels were high. So then we thought that even the levels of Zeb1 is high, it may not be functionally full. So for that we did some chip PCR to see that since Zeb1 binds to E-cadherin, even the Zeb1 is high, is the binding of Zeb1 to E-cadherin is okay or not? And we found that Zeb1, though it is high, but is it not able to bind to E-cadherin promoter properly, as well as through chip mass spec, we saw that ZEP1 during hyperglycemia condition is binding to some pro-inflammatory molecules like S100A8, S100A9 and all. So that means the functionality of ZEP1 is lost. And that gave us clue why even ZEP1 is higher, it is not able to close the wound properly. So one of the functional thing that we did, we tried to knock down this ZEP1 during hyperglycemia condition. And then we saw that yes, inhibition of ZEP1 in hyperglycemia can actually rescue the endothelial cell function through over here tube formation. So these two things then also led us to see that what is the effect which is causing MADE200B elevation during hyperglycemia and how can we rescue that. So for that, we looked into the epigenetic regulation of microRNA 200B during hyperglycemia. So I will just tell a few things about how we do methylation study. So suppose there is a gene and it is the promoter and it has a CpG site which can be methylated. And methylation means gene suppression. But by any mean during any physiological or pathological condition, due to the action of some DNA methyltransferases enzymes, this CpG can be unmethylated, and then it will lead to more gene transcription. And then this is the same thing that was happening for MADE200B in condition of hyperglycemia, that the promoter was getting unmethylated. So here, these all dots means one CpG that was studied through bisulfite sequencing. And here in hyperglycemia, you can see some patches of the CpG islands that were unmethylated in case of hyperglycemia. And that is causing to the upregulation of microRNA 200B during hyperglycemia. So if microRNA 200B is being regulated by hypermethylation or hypomethylation, then if we can reduce the microRNA 200B levels by providing some DNA methylating agent, then we can rescue the wound. So this was our hypothesis. For this, we tested a molecule called S-adenosylmethionine, and it is a DNA methyl donor. So what it does, it can then we saw in endothelial element, it can reduce microRNA 200B, and this was associated with elevation of wound perfusion. That means if by any mean you can methylate microRNA 200B promoter, and then you can lower the microRNA 200B levels, then you can rescue the wounds perfusion as well as healing over here. So in SAM treated animals, you are seeing the wounds were healed compared to PBS treated diabetic animals. So with this, I will conclude my first part of my talk, and I will say that microRNA 200B inhibits wound angiogenesis, and ZEB1 increases wound angiogenesis by reducing microRNA 200B in normoglycemia condition. But during hyperglycemia, ZEB was dysfunctional, as well as it was not able to control MADE200B. MADE200B levels were high, and then wound angiogenesis was stalled. But by any mean, if we can methylate the MADE200B promoter by any compound like S-adenosylmethionine, you can rescue back the wound angiogenesis. For this part, these observations were published in two different papers, one in diabetes and one in molecular therapy. And this was like all the work I was doing with my group in my postdoctoral training period. So now I will switch to my second part of my talk, and what is I am doing right now, what is my group is doing right now. Okay, so I'm working under the umbrella of Indiana Center for Regenerative Medicine and Engineering. So we are looking to how to regenerate tissue, organs in vivo, not in vitro, in vivo, and how to rescue any tissue degeneration phenotypes. So I will start with explaining what is regenerative medicine. So it is a collection of techniques to restore our physiology to something that resembles its original condition. It might not be 100% original, but it should resemble original condition. And reprogramming is one of the method of regenerative medicine in which you can convert one somatic cell directly to another somatic cells. So suppose if my endothelial cells is lost, I can try to reprogram fibroblast or other cells which has some reprogrammable capacity to serve like endothelial cells. But there are a few challenges. First thing, that if we want to reprogram in a live condition. Otherwise, what happened if you reprogram in petri dish and put it into an individual, there must be immune rejection. So for a good reprogramming method, it should be under immune surveillance. So how it was done, and maybe many labs are doing right now also, that they take the skin fibroblast or any cells that they want to reprogram, they expose them to nuclear reprogramming factors, make it to pluripotent cells, and then they will take to what cells they desire. For example, this example, they are making fibroblast into neurons through induced pluripotent cell, and then they can using this in cell therapy or studying the disease model. But in this condition, in this reprogramming, we have seen that the reprogramming efficiency is not more than 5 to 10%. So then what is happening to other 90% cells which are not getting reprogrammed or which are getting very less reprogrammed and not giving into your desired cell phenotype? So one of the main consideration can be they can form cancerous cells. So then to circumvent this problem, scientists came with the idea of direct reprogramming, in which they can reprogram one somatic cell directly to another somatic cell without going through this pathway of iPSCs. But here also we were using viral particles, and we know that they have certain possibility to lead to cancerous cells. So our group, I was fortunate enough to join this group in my postdoc, which was trying to do a topical tissue nanotransfection. It is a method of delivery of plasmids or anything in a non-viral condition, non-viral stroma. We are trying to reprogram the cells and trying to rescue the disease phenotype. So in this example, in this paper, we developed a chip which is this much of size, and then it has some pores which can electrophoretically deliver any charged molecule. For this example, we delivered plasmids, DNA, which are negatively charged over here. And then through apply of a focused electric channel for a very small time, less than a second, you can deliver plasmids to the epidermis or dermal region, and then you can try to reprogram the cells. And this is one of the validation that we did, that yes, our chip is working. So I will show you one or few examples of what we did with this. So this is TNT is basically a delivery platform, and then you can choose your cocktail based on the literature, and then you can try to deliver and see how it is going. So in this example, we took a cargo called EFF. It is a combination of E2V2, FOXC2, and FLE1 in 1:1:1 ratio. And we used to rescue the hind limbs. So in this, we used a hind limb ischemia model in which we transect the femoral artery so that there is no blood flow in this limb. And then at day 3, we try to provide the vasculogenic cocktail to try to reprogram fibroblast lying over here to endothelial-like cells. And then we were succeeded in this thing that at day 7, you can see in TNT EFF treated samples, you are trying to rescue the limb where the limb was not perfused in TNT control subjects. And this is the measurement of the perfusion over here. Even you can see the limb over here in one mice model, BL6. And then there is another mice model over here in which if you don't provide the vasculogenic cocktails, your cells are like, your limb is like this, but you can rescue the limb if you provide TNT EFF. So now our group is trying to work on this and trying to use this TNT technology in different disease models. For example, in ischemic stroke, in diabetic polyneuropathy, in peripheral nerve disease. The only thing that we need to do is to change the cocktail as per our need. So for example, for ischemic stroke, they have tried itovitu, FOXC2, and FLE1. For neurogenic neuropathy, they have tried the other cocktail called ABM. So it's like you change the cocktail, and then you can try to reprogram the cells under live body. So then I am using this technology for CRISPR-Cas9 mediated gene demethylation. So as I mentioned in my previous talk that in diabetes, there is a lot of DNA methylation and demethylation events going on. So if I can specifically demethylate or remethylate a gene promoter, then I can rescue that gene phenotype. So for this, we have used CRISPR-Cas9 approach. So Cas9 inherently has a cleavage domain. But by engineering, if you can deactivate that cleavage domain, it will give rise to dCas9, which have the specificity to bind to a given gene promoter gene based on the guide RNA sequence. And if you can bind an effector domain, for example, if you want to do a gene methylation, in this effector domain, you will have a DNMT, DNA methyltransferases catalytic domain. If you want to demethylate, you can have in this effector domain as TET domain. If you want to do histone acetylation, you can provide the catalytic domain of a HAT. So based on what you need, you can change this effector domain, and then you can do whatever the gene manipulation you want to do. In this case, you want to do epigenetic modification. So for example, if there is a methylation group over here and I want to demethylate this group, I will change this catalytic domain with a TET effector domain. So that will go and demethylate this region. So for example, this dCas9 will sit with the help of a guide RNA, and then this will try to demethylate specifically in your gene of interest. And then you can also achieve the specificity for cell type selection. For this case, for example, I want to do epithelial mediated DNA demethylation. So I will choose a guide RNA which is driven by a promoter KRT14, which is expressed in epithelial cells. If I want to do in endothelial cells, I will just change this KRT promoter to any endothelial specific promoter like CDH5. And then I have plasmid dCas9, and then I have the TET1 complementary like CD, the active site of TET1. And then through TNT, I will deliver these plasmid cocktail to any mice model, and then I will validate. For example, in this case, if I used KRT14 driven. So then I will see that how much in KRT14 positive cells my plasmid of interest is there. So this plasmid was GFP tagged. So I will measure like around 75% of keratinocytes were containing this plasmid. So this will gives you the specificity of the plasmid. So for this, as Dr. Kapil mentioned, that I recently got a grant from US Department of Army, and the title of this grant is Targeted Epigenetic Gene Editing in Vivo to Rescue Diabetic Ischemic Wound. But frankly, it can be used for any kind of gene editing, epigenetic editing through our TNT. So this is the thing that I am doing right now. And then I will be also explaining one technology that my lab is right now doing. It's called single cell RNA sequencing. Okay, so first I will tell you the need of this technology. So we all do RNA-based analysis. So suppose in vivo setting, we are doing a bulk RNA analysis that we generally do. So if say we have two cell types, fibroblast and endothelial cells, and if we are doing bulk RNA estimation, so we don't know which fibroblast or endothelial cells, which gene expresses what level. For example, if a gene is elevated 5 folds in fibroblast, but it is downregulated in 5 folds in endothelial cells, in bulk RNA technology, you will see +5, -5 effect will be 0. So you will not see any effect. But in single cell, you have ability to go and see the dynamics of RNA expression in each and individual cells. So for example, if one cell has this much of expression and the last cells have lesser expression, you can categorically say that, okay, in my fibroblast, this gene is expressed how much, and in my endothelial cells, this gene is expressed how much. So this is the power of single cell RNA-seq that you can do. The assay design is simple, like we do all the time. Like we will take the tissue, we will digest it with enzymes, different enzymes, to make single cell RNA single cell suspensions. You will also remove the RBC because RBC will not give you proper RNA outputs. You will also remove dead cells. So one of the important consideration for this is you should not have more than 5 to 10% of dead cell in your suspension. Otherwise, when you do RNA-seq analysis, you will not get good results. And then you will proceed with single cell RNA-seq. This is the platform. It's called 10x Genomics. Right now it is around like 400 to 500 boxes over here, but I know in India also they have started. So I will just explain in a bit what it does. So suppose there is a one cell, and if you can encapsulate that cell into a like oil bubble, and in that condition, in that sphere, if you have say a barcoded gem, it's called gem over here, or gel beads over here, and then you have RT enzymes and other things. So what will happen? The cell will burst, and these gems will provide a marker to that cell. So that means all the mRNAs of a particular cells will be tagged with a particular barcode, and then you will have 10,000 of different cells with 10,000 of different barcodes. So that means every mRNA will be tagged with cell-specific barcodes. And then when you do the analysis, you can see what cell is what. So for in this example, in a wound samples, the single cell isolated cells, we can cluster different cells based on their markers. For example, cluster 0 over here is fibroblast because it was very highly positive for classical fibroblast marker like collagen Or if you are saying that this is myeloid cell, then it should be highly enriched in myeloid cell marker like lysozyme and everything. So good point is that you can specifically take out the myeloid cells through bioinformatic analysis and see the gene expression pattern. So then it will mask all the heterogeneity from other cells, and you can only study the cell of your interest. So suppose you are saying that your treatment is affecting myeloid cells. So then you will, in an in vivo setting, you will isolate only the myeloid cells, and based on your condition, like control and treatment for here, I have wound and severe wounds. So then you can distribute your myeloid cells based on your condition and do the differential expression analysis. So over here, for example, this all are individual cells, and these all are individual cells for other conditions. So if you are saying if a gene is highly expressed, that means you can count, or you can see how many cells you are having the same phenotype, like high expression of that gene. So this is a very powerful technique, and we are trying to use this technique in all our reprogramming events, because we know that when we reprogram, not all the cells will reprogram. So that means then we are calculating, okay, how much, if we are saying we are converting fibroblast into other cells, so we are now looking at how much of those fibroblasts are getting converted into endothelial cells and how much are not getting converted into endothelial cells, like that. So with this, I will conclude my talk with acknowledging my mentor, Dr. Sen. He is the director of Center for Regenerative Medicine and Engineering, where I am working with. And he is a good orator, he is a good thinker, and thanks to him, I'm now starting my own laboratory over here. Past 2 years, I'm working, and now thanks to DoD, I will be working on an individual project. And this is my group. This is pre-COVID times, and I like this photo a lot because now we are only meeting on Zoom. So with this, I want to play a short video. So I will stop the sharing now, so that.
Yeah, so I can share I can play the video.
Yes.
Yeah, so give us a minute.
Yeah. So okay, Kapil. So this is about our technology, TNT, and you can please see this, and this has implication in different. We are not the sound.
They acquire specialized functions. For decades, scientists believed this process ended with terminally differentiated cells. Committed to a particular cell fate, the ability to switch function irreversibly lost. In a major breakthrough, iPSC technology established the possibility of inducing plasticity to terminally differentiated cells. But several limitations arose, including a lengthy multi-step lab-based process, possible inefficiencies due to experimental conditions, low fidelity with which the final cell type is generated, and safety concerns, including potential cancer risk. More recently, scientists found that mature, fully differentiated somatic cells could be directly converted to another cell type by ectopic expression of specific transcription factors, without the need to acquire intermediate pluripotency. However, this technique requires viral gene transfer, an approach which, 1, requires an exhaustive laboratory procedure, and 2, carries risks, such as insertional mutagenesis, irreversibility caused by stable transduction, and increased cancer risk caused by long-term proto-oncogene expression. Therefore, it is not well suited for point-of-care applications. Thus, a non-viral tissue nanotransfection approach was developed for direct cell conversion in vivo by Professor Chandan K. Sen in collaboration with Professor James Lee. This work has been published in Nature Nanotechnology in 2017. This approach is simple, does not require elaborate laboratory procedures, and can be used as a point-of-care approach. This approach consists of two components. First, a hardware component, or chip, fabricated for efficient delivery of cell-specific cargo in murine model. Manufacturing of this chip has been scaled up to fit dimensions relevant to human need. Such chip has been successfully tested in preclinical porcine studies. Second, the cargo itself, which consists of a combination of different reprogramming factors. Thus, by applying a highly intense and focused electric field through arrayed nanochannels, this device operates the adjacent tissue cell membranes and electrophoretically drives reprogramming factors into the cells. This new nano-electroporation technology is distinctly different from bulk electroporation. The simplicity and utility of this approach was demonstrated by nano-electroporating the skin that served as an agricultural land to cultivate cells of different functional abilities. Using a specific cargo, a combination of different reprogramming factors, skin fibroblasts were successfully converted to endothelial cells that ultimately formed blood vessels with 7 days. These blood vessel networks anastomosed with the parent circulatory system and restored tissue and limb perfusion in 2 murine models of injury-induced ischemia. Similarly, using a different cargo, skin fibroblasts were converted to neuronal cells that showed similar gene expression profile and electrophysiological function to that of cells in the developing brain. This tissue nanotransfection platform technology has the potential to ultimately enable the use of patients' own tissue as a prolific immunosurveilled bioreactor to produce autologous cells that can resolve conditions locally or distally upon harvesting. This simple-to-implement nano-electroporation technology generates favorable biological responses. It is a one-time treatment applied topically and only lasts for a fraction of a second, without elaborate laboratory procedures. And it could have applications beyond reprogramming-based cell therapies, such as gene, protein, and drug delivery.
Thank you, Kapil. So as I mentioned, now we are trying to implicate this technology for drug delivery and other like gene expression analysis. So you can provide shRNA to knock down the gene. You can provide ORFs for open reading frames to increase expression of genes through DNT. I am using for epigenetic editing of the tissue. So all the things. And good part is that we have an in vitro component of this. It's called nano electroporation, NEP. So you can do a mechanistic study in those technology also. So with this, I would like to thank Dr. Seth and other organizing committee to invite me for this. Thank you. And I'm ready to take any questions.
Thank you very much, Dr. Kanaya, for your insightful talk. Please say our greetings to Professor Chandan Sen from my side and from NIPER Guwahati. So particularly inspired by your group, your team. So recently I have started working on this wound healing scaffold with a couple of very much young, enthusiastic and dynamic students. So in particular, my PhD students, Stibha, Sara and Mohit, they are eager to explore this field. So yeah, we love to collaborate. So trying to synthesize some small molecule scaffold. And thank you for accepting our invitation, because I understand it is pretty much early morning in India. And yeah, it may be a little bit tough for you and at this particular in this winter season. So thank you very much. And now the session is open for question answer. Dear participants, if you have any question, please feel free to post into the chat box. So, sir, dear participants, if you have So I think there is no questions from the participants. So dear participants, if you have any question in future, please feel free to email Dr. Kanaya. He will be more than happy to give the answers. And thank you, Dr. Kanaya. Thank you again for participating in this event. Thank you very much. And yeah, now slowly we have moved to the valedictory session of this event. So NIPER Guwahati is grateful to our chief guest, Professor Govardhan Mehta, and all the speakers for accepting our invitation to deliver the talk. We are pretty much thankful to the faculty members, staff, and Computer and Communication Center of NIPER Guwahati. Special thanks to Professor Murthy, Dr. Amit Alexander, and Dr. Krishna Undela for their constant support and encouragement constantly from the beginning of this initiative. And finally, I would like to thank all the participants for making the event most successful. And thank you very much once again. So dear participants, so you will be getting the participation e-certificate shortly. Thank you. So now it's time to conclude, and thank you all. Okay, Dr. Kanaya, thank you.